every community meeting. And between 2015 and 2016, we've had these working groups meeting, every other week. And the amazing thing for me has been how cooperative and how hardworking everybody has been doing this work, and really for a common goal, to try to see how we can facilitate sharing and comparing these data. So I've really been impressed by the cooperation and the working for this common good. So I have a few housekeeping things. We have the meeting during the day, which is sponsored both in terms of the facility and travel for the speakers from the NIAID, both the Division of Allergy, Immunology, and Transportation and Division of AIDS. So I wanted to thank the list of people helping meeting support: Grace Tellini-Farrell, Megan Curry, Marlene Goldman, Stacy Gray, Jasmine Hunt. It keeps getting more and more. And then in the evening, we have demos of tools and resources back at the Hilton, and then also supporting the travel of early career researchers. We have our industrial sponsors, Amgen, Antibody Science Society, Genentech, Grifols, Illumina, and TP LabTech. And the Ermac Center at Simon Fraser has helped a lot with logistical support for the meeting. And then just to introduce the organizing committee. So these people have been working on it for 2 years, as I say. We've got myself, Felix Braden, Danny Dewick, Tom Kepler, and Jamie Scott. And then with the organizing committee, we have 3 working groups. We have 2 of the co-chairs of the working groups have joined the organizing committee. So Common Repository was Lindsay Corey and Lindsay Cowell and Corey Watson. Minimal Standards is Steve Kleinstein and Nina Prack. And Tools and Resources, Eric Mattson and Chaim Schramm. And then a few details. We have the boxed lunches, and people with non-U.S. credit cards were having trouble, but that should be straightened out. And so boxed lunches will be for the poster sessions tomorrow and Wednesday, and that should be accepted now. Evening demos are in the Regency room in the Hilton, and we have a cash bar, which sounds cheap, but we did that to try to get all that money and support as many early career researchers travel awards as possible. And those people should see me to get the reimbursement forms. If the speakers can bring their presentations on flash drives and give them to Jasmine in the back, then they can be scanned for viruses and loaded up ahead of time. The group dinner is at Carluccio's on Wednesday night, and there's directions in the packet. And if you email me by tonight, I can adjust the number of people for the Wednesday night dinner. There are a couple others that Grace told me. The internet, no password, but it's just NIAID, and it should be no password. Restrooms are in the lobby area outside the coffee shop. Coffee shop closes at 3 p.m. The restaurant closes at 2 p.m. And then there's a convenience store near one of the rooms, the VIP room, that closes at 4 p.m. The reception can call you a taxi out there. And then there's a closet that can, for luggage, that will be watched during the day. So if you contact Grace again, they can put the luggage for the day in the closet, and that will be safe and watched. And then I'm just going to go over the agenda for this afternoon. We're doing our little intro. And then we're going to have Tom give us an overview of the meeting and goals. And then each of the working groups are going to take an hour. We're going to start with minimal standards, then common repository, and then tools and resources. This is the first time that they've brought their recommendations to the full participants. And then starting at 6, we have our reception for tools and demos of tools and resources back at the hotel. So with that, I've asked Tom to come up. Okay. I'm going to assume that that's going to work.
I'm just trying to see if I can get it to show me the notes at the same time on this monitor. Oh, okay. Well, then we'll do it this way. Good afternoon. I was hoping that I'd be able to use my notes on this, but I can't, so I'm going to be winging it. Let me just start off by saying how grateful I am to Jamie and Felix for having this idea and for getting this community together. It's been a tremendous effort on their part. I had the good fortune of meeting Jamie and Felix at the Antibody Society meeting in 2014, and it was at that meeting that they told me about their ideas for getting the community together. And I thought it was just a terrific idea. And so I'm grateful for the opportunity to be working with them and to be here addressing all of you and working with you as we move forward in this ambitious initiative. So we're all here because of the promise of technological innovation. Technological innovation allows us to see things for the first time, things that had not been seen before. And I believe that we are on the verge of seeing things that have not been seen before in antigen receptor repertoires. There's an enormous amount of information contained in the adaptive immune system, and we're only on the very edges of understanding what that is. Galileo looked through his new technological innovation and saw for the first time the moons of Jupiter, including Io, Ganymede, Callisto, and Europa. And after Friday, I can barely say Europa without a little catch in my voice. It was such a good idea. And we're here because we are a community. I myself was trained in immunology. No, I wasn't. I was trained as a physicist. And I learned as a physicist about the existence of these things called B cells. And I learned that they had genes unlike any other genes known on Earth, that they were assembled at random, and that B cells went through a Darwinian process of affinity maturation. I thought this is the most extraordinary thing that I ever heard of. And so I learned immunology, and I haven't looked back. We all come from similar diverse backgrounds. We're physicists and computer scientists, immunologists, doctors, and many other things as well. And we understand that in today's science, more than ever before, we are absolutely dependent on each other, absolutely dependent on the technologies and on the methods for analyzing new technologies than our predecessors were. We can no longer carry these things out by ourselves. And so the whole point here is to make this community stronger and to allow us to share with each other the insights that will make each of our research programs stronger. One of the things that I wanted to describe was lessons from the past. We were all familiar with this phrase, those who cannot remember the past are condemned to repeat it, coming from the mouth of George Santayana, who's a great philosopher and poet. And although I'm not going to be telling you about the great promise of this technology, we're here because we already believe that. I'm not going to proselytize for it. I'm not going to be in front of you an evangelist for repertoire sequencing. What I am going to do is to remind all of you of a lesson from the not so distant past of immunology. And that's this story. Please forgive me for bringing up hurt feelings and causing offense potentially, but I'm absolutely fascinated by this era in immunological history. For those of you who have come to immunology too recently to understand what this is all about, let me tell you very briefly. In 1971, it was demonstrated that immune tolerance could be transferred from one mouse to another. And this was an extraordinary finding and one with a huge amount of potential for future research. And indeed, that topic was taken up and pursued by a large number of immunologists moving forward from that. And in fact, I guess is there a, I'll point and you'll be able to see where I'm pointing, no doubt. The figure here shows the number of citations for two different phrases. The first phrase is regulatory T cell, and the second phrase is suppressor T cell. So these are the number of citations per year for suppressor T cell, and this is the number of citations per year for regulatory T cell. Oh, now I've got two of them. That's great. I have to, well, anyway, yeah. Well, this is actually does, I will tell you, this reminds me, I was sitting, I was giving a talk at a conference, and there was an older gentleman, a very distinguished man from a European university, who was sitting in the front, and he was chair of the seminar, and I was giving my talk, and I kept getting confused by this second light that was going by. I'm thinking, what the hell's going on? And I look, and it's my distinguished colleague from Europe who's throwing me off. Anyway, so this was an important phenomenon, recognized as such, and it attracted a lot of interest and a lot of funding. And it grew very quickly over the course of the next several years. Included in that time was the mapping of the determinants of this suppressive activity to the IJ locus of the mouse MHC. This mapping was done by very, very clever and very time-intensive, labor-intensive task of crossbreeding mice using mouse genetics to understand the MHC locus. At that point, we did not sequence DNA, and everything that we knew about the MHC was what we knew by classical genetics and these extraordinary mouse strains. This was a new technology, and I think you know where I'm going with this. By 1986, 10 years later, we did have DNA sequencing, and when we sequenced the MHC locus, lo and behold, the IJ region contained no genes. And it was at this point that the suppressor cell story began to fall apart. To tell you the truth, it had begun to fall apart a little bit before then, but this was the proverbial stake in the heart. Now, some years later, more discoveries were made, and a new research program was begun on regulatory T cells. And strikingly enough, these cells had the capacity to provide tolerance. They could be transferred from one animal to another, and for all the world looked like suppressor T cells, but it was very difficult to get this work going because of the stigma attached to suppressor T cells. And there was a lot written in these years about whether suppressor T cells and regulatory T cells were the same thing. And the point is that mistakes that were made early on had a very disruptive influence on subsequent research. So I think that it's really incumbent upon us to be very careful, to understand exactly what it is that we're doing and to validate our research. And importantly, these concerns are not our concerns alone. These concerns have risen to the very highest levels of scientific research, like The Economist, where they publish, and I mean, actually, I mean that seriously, because it's the people in places like The Economist that determine what money is going to be spent on what. And there is a great deal of concern about replicability, reproducibility, and so forth, the reproducibility crisis in science. Well, whether or not you think that it is of the magnitude that is discussed in the pages of The Economist and the New York Times, it still is the case that we are beholden to the people who fund our research. And I suggest that one of the key reasons for having this meeting is for us to step up and take responsibility for that research, for validation, and for making sure that what we're doing comports with the overall goals of science. Because if we don't, it's going to be imposed upon us. So what are the goals of the meeting? So the goals, as quoted from last year's report, the goals are to develop action plans and recommendations that will be used to make Repertoire NGS data production, analysis, and sharing across academia and industry as seamless as possible, while recognizing the rights of and responsibilities to the donor communities and taxpayers. This is the charge that you are all given. We're here to really organize ourselves and to make these things work. Our goal here is to facilitate research, not to impede it. My Debbie Downer introduction notwithstanding. To accomplish that, as Felix has already mentioned, the community has organized itself into 3 working groups: the Minimal Standards Group, and the charge to the Minimal Standards Group is to determine minimal metadata to include when publishing AIRR data or submitting them to a public database. The Tools and Resources Working Group is charged with the development of common biological and data analytical standards for validating experimental procedures common to members of the AIRR community. And the Common Repository Group with developing platforms and platform criteria for facilitating sharing of data among researchers. And this is important. In 2015, the meeting was called to determine what the issues are. What do we need to discuss? What do we need to come back to? What do we need to organize around? And that, I think, was accomplished. The issues have been described, and they are going to be discussed at length here. The action items for today and for the next 3 days is to delineate the actions that need to be taken for us to address these issues. And in particular, we want to write and ratify the recommendations that will be shared with publishers and funding agencies and others. We are going to want to disseminate these findings to the larger research community. And last, but not least, we need to plan for the future of the community effort moving forward. And so I won't belabor the point any further. I'll just ask you to go forth and make awesomeness. And we'll turn it over now to the workshop leaders.
Thank you, Tom. I knew you'd do the job. And yeah, we're first going to hear from the Minimal Standards. The coordinators were Steve Kleinstein and Nina Crack. Okay. Yeah. Yeah, I did want to point out, so we have a pretty good registration through our webinar, and this is all going out. And Eric has been kind enough to monitor that, and so if there are questions, he's going to bring them forward. But also, we now have a request that if you could use the mouse for your pointer, then the webinar will be able to see that.
Okay, hi. I'm Florian from Stanford. And yeah, I will talk today with Sam a little bit about what the minimal standards are, so what we are coming up for a recommendation, but it's, as said, it's a working group and we are not completely done. This means that we have proposals. We met quite a few times online and in person, and so we would like to, or I would like to ask for your contribution on it, so what you think about, if it makes sense what we're doing or what we're thinking. This is a working group created last year after the meeting in Vancouver. And if we miss someone here, so please let us know that we can add you, and everyone is welcome, of course, to join and push it forward. And what was the task of the working group is to really identify the standards, what has to be reported or what should be reported of data submission to a database as well to journals. And because, as said, it's still a quite new field, and a lot of things got different ways, so everyone was experimenting what is the best way to do it for themselves. But now I think for a certain part, and we will come to it, there come some standards. So these are important things to know if you want to know how the experiment was done, if you want to redo it, if you want to reanalyze the data and compare them with other databases or with other data sets. And we also developed a lot of standards or terms, and we compared them with what is already existing in different databases like dbGaP, SRA, IDB, import, JServer, Receptor, AIRR. So because a lot of terms are covered in different ways, and we wanted to get an overview what is already existing and what is new. And one part which is more or less good covered is especially the sequencing part, how the sequence is generated and everything. And we think it's important. And yeah, as we said, we were meeting a lot of times, and the result is that we have 6 high principles with subpoints. And we working together, or we will work with the existing data repository, so with a working group of databases to bring them alive. And so, as I said, the last meeting was to bring this up, to develop these terms, and we did this. And the new field is, so a lot of people are here from academia, or nearly all, but also some companies started to do sequencing. And this is a good thing because it's also standardized ways to do stuff, because the company most of the time sticks to it. But also they do it sometimes differently, and they also not release all the information how they did it. And one question is what kind of information you would still like to get from them, and how much we really need to reanalyze the data or to quantify if it's still a good job, is the data actually usable. And then we come up with a checklist with a lot of different terms with explanation. And the question is also to this community, is this enough for a minimal set, or should we add something or change something? And yeah, what is the scope of the standards at all? Short reminder for what did we need is actually for the data generators, the wet lab part, which could be individual labs, core facilities, or companies, especially core facilities. I'm from Stanford, and they're looking for some minimum standards to tell them, so, okay, this is all the information we should provide, this is the way we should cover what we're doing, which is also at the end usable for journals, so that we really make sure that we sequence the stuff in a good way and we report it in an accepted way, in a community accepted way. And the researcher should also know from the beginning which data he should collect, which he can maybe easily collect in the beginning, but if you don't do it, then later on it will be much harder. Then also the burden of deposit your data in a database is sometimes easy, sometimes a little bit harder, and we would like to streamline this process by streamlining the whole data generation. So if it's a lot of created in the beginning, then it's also the uploading is much easier and faster. And of course, it's the journals will ask for certain information, and we would like to give them a list of what we would recommend that you should ask as a journal and what we will do with it. Data processing is reanalyzing the data, and also if there are standards out, then you can build the pipelines much faster and better. And you can also compare the data sets from different databases and or from your own lab and different labs. And you can reuse pipelines for it. Yeah, which data should be restored? And coming to the 6 core elements, so from now on, I would like to get some feedback. The biggest part is the study, sample, processing. This is, a lot of these parts are already covered, the study and sampling, with existing databases. This has nothing to do with repertoire sequencing, but still we would like to encourage that we need information about the studies, the subject, so is this mouse, human, its age, gender, and also if we can, with ethic issues, we would like to know about what the people have. If it's human, so if it's, is he, she's sick, so what kind of diagnosis he has. We'll come to all this in detail, but just to start first. The sample, those cell types is important. It's not only that I sequence B cells from everything, it would be also nice to know if you made a subdivision or subsampling, then what have you used for it, then how you process these cells, and then especially now becomes more the sequencing part, how you generate actually libraries. So starting from the RNA, DNA purification or from single cell, and how you sequenced it, and then coming to the FASTQ file is the output file from the sequencer. So until here, then if it's done, then you can't change anything anymore, but this is the output file from the sequencer. The processing is the first processing, annotate or align read 1, read 2, what cutoffs you use for defining primers, and all these is not the VDJ assignments themselves. So this is the step before. So the output from the processing is a FASTA or FASTQ file, which can be used or will be used for the VDJ aligner to assign the V and D and J segment to it. This is, we stop where the VDJ repertoire actually get assigned. The major thing for this is that we think everything after, there are a lot of different versions out, and so this is up for discussion if it's the right point to stop. But we think that this is, until here, we have a good agreement what is standard. So even 5.5 is, you can use different cutoffs for the primary assignment, but it's not that arguable. And everything before 0.4 you can't change anyway. So this is set up in the how you set up the experiment. Okay. Going a little bit more into detail, so core element, yep. Sure. Yeah. Yeah. Is yeah, 4 core elements to really define, but stressing the point that we really would like to have the FASTQ file from 0.4 that is get stored as well. It would be nice, or we think it should be that also the FASTA, FASTQ file for the VDJ alignment should be stored. This is up to question how or where, but yeah.
So could people who are asking questions use these 2 microphones? We could have somebody doing that. Again, because we've got like 20 people in on this from afar. Sorry about that. Otherwise, could you restate the question and maybe not have too much of a discussion? Right? Yeah.
So I guess the question was whether there are 4 or 6 key elements that we're proposing to look at, and I guess there are 6 key elements, I guess, labeled in the plot up there: 1, 2, 3, 4, 5, 6. 2 of them are essentially sequence data files. So 4 and 6 are data files, and this is really sort of the crux of the standard. Something that we was a decision that we had to make as part of the minimal standards working group. Sort of at what level were we going to recommend that people, when we publish our data, when we deposit it in a repository, what exactly are we going to have suggest that people deposit with their publication? And there has been quite a bit of debate within our working group and with the other working groups on this question. And the decision we made, and we listed on the slide and for mention very explicitly, is what we think you ought to deposit are essentially two main data files along with supporting information. The one data file is the raw FASTQ sequencing data. That's element 4. And then the second is this element 6. It's the sequencing data file that contains the immunoglobulin receptor sequences where either the paired end reads have been paired, the unique molecular identifiers have all been collapsed into consensus sequences. It's sort of at the point where you can start to assign germline VDJ segments. And so the recommendation is that before you do that process of assigning the VDJs, before you do the process of identifying clones, that's the state of the data that we would like people to submit with their publications.
I'm sorry, can I ask a quick question? But why don't you want to also include the VDJ assignments that the person is saying that they are?
So there are a couple reasons. One of the main ones is that the field of data analysis is evolving very rapidly. And so our fear with creating standards is that we worry that the standards will be out of date by the time we've published them. And we don't want these standards to really be things that stifle innovation. What we want the standards to be is a framework for describing an experiment rather than dictating how the analysis work should be done per se. So that's one reason. A second reason is that there's another working group that deals with tools and analysis methods, and so we thought from the minimal standards perspective of describing the experiment that our task sort of ended at the point where we have data that go into a repository, and that the analysis piece of the standards will kind of take on at this point. So those were our main reasons.
Yeah, so just to elaborate on that, we do think people ought to report, in the ideal world, people ought to report the VDJ germline assignments. People ought to report the clones. We would encourage people to do that. We don't think that ought to be required as part of the publication. That when you submit the data, when you publish in a journal, the journal should be requiring this level of data be put in a repository.
I think also one of the challenges with mandating certain types of analysis methods is because things change, not just the analysis methods, that there's also what you're comparing your data to. So if you want to try to compare the clones that emerge from one study to clones that emerge from another study, and you want to compare them to a common framework, you may wind up reanalyzing the data anyway. And the alleles that you use for comparison, so what database do you use to assign the germline alleles, for example, that will also change with time. So these things are very tricky, I think, to codify in the same way that the rest of these standards could be codified.
But we believe this may be one of the most controversial statements we're going to make. And it's a question.
Well, so the goal of the standards is as a descriptive way of capturing data about an experiment, so that when somebody analyzes the data and withdraws it from a database, they can make judgments as to the adequacy of how the experiment was performed. So the goal is not to dictate terms to the experimentalist about the level of quality required or the number of replicates needed to adequately sample a population for clonal overlap or things like that. It's really to capture a complete or adequate description of an experiment so that the data can be interpreted and compared between different databases, potentially in different experiments.
And maybe also to rephrase it a little bit, I mean, the group is called Minimal Standards, but it's actually about minimal information. we capture information, we don't set the standards how you should do your experiment. Just what kind of information we would like to have to be able to reanalyze it and to quantify what you actually did. Yes.
Perhaps it's something that's covered by another working group then. But I do think there's a crying need to give some advice, not standards, but some advice to people doing experiments about what's expected. And I mean, we're all people that could end up refereeing a paper tomorrow on repertoire analysis. So, you know, one of the things I think we ought to be able to say is, what would we expect? Not from somebody that's doing groundbreaking work with new experiments, but from somebody who's working with the state of the art today with Lumina sequencing, say, you know, what level of quality do we expect? You know, how do we expect the data process so that FASTA file is in a good state to move on to the next step? Is that covered by another working group, or would it be with you if it was something?
We don't cover it. We don't make recommendation what is a minimum quality or what is a minimal RSQ base to be a base called a base. We don't do it. So this is, I'm not sure if we can do it or if we should do, but we would like to have the information to make some judgments about how the procedure actually did. And I think the whole, maybe a little bit in the direction, I think everything until step 6 gets more and more streamlined. So this, therefore, is also companies doing it, is kits coming out to do it. And yeah, maybe at some point there should be standards defined, but it's not, we don't do it. Not our group.
Well, as I said, I think it's guidelines rather than standards. And I'll just leave it out there then as something personally.
Yeah, maybe we can develop for next year.
Well, actually, there's one thing that, you know, the working groups, putting some of a little bit of what everyone's doing together, maybe at some point, it might be a good idea to put together a document that goes from the very beginning to the very further, you know, kind of the end analysis of, let's say, clonal expansion and clonal lineage, and walking people through that as an example of where some of these things fall. But it would also help people when they're new to the area to walk themselves through those steps, you know, and be able to say, you know, that what the scoring is on the sequence calls and all that, and how to deal with that. And that could even refer then to papers on how to do that, for instance. But it would all be by example. And so we don't have, nobody worked on that full soup to nuts description. But it might be good that if some people here wanted to work on something like that, that they could, in the end, and incorporate some of these recommendations and guidelines that are coming out of the different working groups to kind of put everything together. So that it's just an idea, but maybe that would be a helpful thing to do at some point.
Just one more point. So related to what everyone's saying, I also think that will help with this idea of reproducibility. Because if you don't give people guidelines, then how do they know how the data is reproducible? I mean, we could talk forever about reproducibility, but I think there's lots of reasons for it. And some of it is, I think, just lack of information, right? Or different systems or whatever. So anyway.
Can I perhaps just get clarification on, I'm not sure how much discussion should take place today and how much takes
Lots
place.
of discussion.
Okay, well, yeah, I mean, there'll be another session, of course, tomorrow on this topic. But okay, if we focus on element 5, we could argue for hours over that point. You know, what sort of filtering is part of that processing before you reach VDJ assignment? I mean, to mention just one, I think identification of chimeric sequences should be a routine process. It's not a process that's often mentioned, but I think it should be. and there will be other people here who will have their favorite technique that they think needs to be applied.
I agree, and I think the goal of the standard is that when you publish one of these data sets, you ought to describe the set of analyses like identification of chimeric sequences that you did in going from the raw data to the processed data. And that's really our goal of this. When you publish these data sets, you ought to describe those processes in a sufficient level of detail, and that's what we're trying. That's what we're trying to capture. And we haven't actually shown you any of the detailed data elements that go into this, but we have them.
We're about to show.
And actually, this was, I would say, many hours of arguing over the details in many teleconferences to get to a set of individual data elements that implement these six.
I'm sure you have, and I guess, well, in fact, Tom's comment about wanting to facilitate, not to prevent research, I think was really critical. But a part of that is to make sure that if someone has identified the 6 core elements, that you don't actually give the study the stamp of approval.
It's not necessarily a perfect study then. Yes, it's just we have the information to know how the study were designed and make a call if you want to reanalyze the data or if you want to use the data for your own study. And if you have the information and you say, okay, I totally disagree how you did it, then okay, I don't use it. But the first step is to have the information, and the next step, may another year or something to develop some guidelines what qualifies a good study. But I think right now it's really hard to say what is a good study. I think we can have some cutoffs, but it's totally depend on a lot of things. I mean, on the how many cells were used, what is the quality of the cells, is this what kind, is this human, is this a cell culture, is this what kind of instrument it is. I mean, I expect a different quality if it's Oxford nanopores or is this Illumina. I mean, this is in different thresholds. But the first step is I have to know, and then we can define guidelines for the most established or common things or standard things to do.
You can also imagine that the guidelines are something that's learned down the line. So once this is implemented, people will deposit lots of studies, and you can just run statistics on what does a typical study look like, what instruments are people using, what are the typical cutoffs people use, etc. So something that can be learned also.
I was just asking a quick question on the opinion of the committee in terms of submitting number 4, the raw data, along with a full description of number 5, so that number 6 could be recreated. And the only reason I ask is because I was wondering what your opinions are, because depositing 4 is easy right now, but I don't think that there's a good framework for depositing number 6 yet. So.
Yeah, this is exactly what we're discussing right now as well. You want to follow up on that?
I think maybe we had a sort of combined meeting of the standards working group and the repository working group, and we definitely got into that a little bit, actually a lot of that. So maybe during that session or at the end of this, we can start to address those questions.
What is the opinion of the committee and users using companies' kits to do the processing, both steps 3, 4, and 5 of their data, where essentially that process is a black box to the users? We don't know how they did the processing. Should we not be using that technology, or should we be working with them to release their proprietary process to the community?
I think it's more the second point where we will go, because also it's, I mean, one reason why we are here is because the sequencing gets more standard. So more people use it, it gets more standard application for a lot of different questions, and the entry barriers get lower. And one reason why the entry barriers to do sequencing, repertoire sequencing, is because companies come in and say, okay, you don't have to know everything, I can do it, this costs you some money, but we can do it for you right away. You don't have to develop something. Nevertheless, I would like to know how they do it. And this is one big discussion point, how much we can force some companies to release some information, or how much we should at some point agree on that if a company, this is a company produced data, we don't know everything. So there are some parts we don't know, but at least it has to be clearly flagged that we don't know how they do it. So I can't reanalyze the data. Just make it then open on public that I trust Adaptive or something, that they do it the right way or they do it their way, and I build on their results. And yeah.
I've just got a quick question about the logistics of this session. Do you have more slides to show?
Yeah.
That's what I thought. So I wonder if we shouldn't go forward with the slides and reserve time at the end for further discussion on the individual points.
Okay.
Yeah, I
Probably should have. Oh.
I have a few questions that I totally agree with Tom should wait for the end.
Yeah.
Hey, but just one question, and I think you mentioned that it was a big discussion, but I think that's a major question that should become before, is the question of stopping at 6 and not going forward. And that has to do with the title of the working group. I mean, is it standards for repository and publication or only for the repository? Because somebody mentioned that many of us here are reviewers, and as reviewers we have issues with what has to be in the actual publication. That's a totally different question than those 6 points, which is what has to be actually given as part of the data to be deposited somewhere. But what has to be the publication, and I agree that it's a different question, but the question if it's part of the working group or not, because if it's not, then take it out, and if it is, then you should go farther than number 6, because number 6 is just the beginning of the publication.
So what we would like to recommend, what we would like to get consensus on as a community, is that when you are publishing a paper that involves this deep sequencing of repertoires, that when you publish the paper, you ought to deposit the data in a public repository, including all of this information. You ought to deposit it in a repository that includes these minimal standards. With the publication.
I probably assumed that we all had looked at the agenda in great detail, and I didn't really go over it. But I just wanted to make a point that, so this is the first time that the working groups will have brought their recommendations to the full community. And then the next 2 days, we get you till Thursday, you poor people. So over the next 2 days, we'll have 4 sessions where each one working group will, again, we'll have discussion, and then we'll have some scientific presentations around this topic. Those are the next 2 days. And then on Thursday morning, we meet as a group again, go through the recommendations. We have a time where we can actually try to come up with consensus for the recommendations. So this isn't the only time that, these questions will be coming up. So again, like with Tom, we need to let them get through there. Well, you're till 5.
That's this one.
Oh, I figured we'd give them an hour, so they should be open around 1:30.
Okay, let's have a look at actually standards. So to the easy part, the core element 1 includes study subject diagnosis. So this is just an overview about the study. By the way, we plan to release all these terms on the homepage so that everyone can have a in detail and the comments there as well. So, but yeah, the study includes who did it, what is it, grant funding, the contact information, everything else, which is quite common so far. The subject is then what we need to know also about the samples, or is it human, animal, what is the age, sex, ethnicity, race is standard in the US, is completely unknown in Europe to report, I would say. Especially for me as a German, it's but there's different standards, and we still here NIH required it a lot of times, so we sort of put it in. But yeah, this is information about the subject and overview. Diagnostic, there will be always ethic issues. How much of information can be released, and can be released in a way that we can reuse the data without much constraints. I mean, we have no, it's good to quantify a study, but we can't reanalyze the data if it's hidden in a secret box and we can't access this data. So there's always we have to figure out what is legally allowed, and it depends on the country as well. So we were thinking about to make a recommendation, and we were stepping away from it because it's just different countries have different rules. But we would like to know something about, if it's a human study, something about the people, the diagnosis, if they're sick or healthy, and if there are other relevant medical history, which is important to interpret the sequencing results as good as possible. The sample, the SCORAMEN2, where's the sample coming from? What type of sample it is? Is this organ, or is it PBMC? Is this a tumor? Is this a healthy material? When is this taken, and how is this taken? Or is it just bought from a company, and then which company, of course? The samples themselves is how is this processed, so the cells most likely, how they are stored, and then the crucial part is how you annotate your cells. I mean, in a way, you should define, when you're talking about naive B cells or CD4 T cell memories, then you should define them, and not only by saying that there are, it's also by using surface marker, like CD4, CD8, and so on. One question is, did we say that we only want the surface marker, or would it be also nice to have the name along? This was last year's discussion as well. One thing what speaks to add the name on it, because it's at the moment the researcher did his research, that is what he was thinking about the cells are. Even it's at this point not true, and or gets later redefined, let's say. But by providing the surface marker as well, then it's all the times can be really pinned down. So this is how the cell type were defined, or is defined. This is the cells you are actually looking at, the sequencing. Yeah. Any comment? No? Okay. Single cell or bulk, I mean, bulk is quite standard. Single cell is coming up much more frequent now, and everything in between, so how many cells is 10 cells or 100 cells, 10,000 cells, millions of cells, and how many cells you actually use in an experiment on per sequencing reaction or per experiment reaction, because it could be also quite informative. How it's processed, the cells afterwards. Are you using DNA or RNA? How's the library generated? Is this a cDNA-based race method? Or is this using oligos to amplify, and that's amplification based on RNA or DNA? Where the primers are located, inside, outside the V region, or frame 1, 2, 3. So all this information would be nice to have, also for later reanalyzing. Are you talking actually about the full construct or just a partial, just focusing on CD3 and all these? What kind of RT reaction is used? And then coming more to how the library for sequencing is designed is how you use barcodes, and where I can find them in the sequencing. Are you using UMIs, the molecular identifiers? This becomes more and more standard. All this information would be nice to have, because afterwards we would like also to be able to extract this information out of the sequencing, to really be able to reanalyze it if we want to. So the primer location, as I said, the sequence of the primers, is this heavy and light chain separated, or are they better, or are they physical linked? So is this one sequencing product out of the both? And how much you use as template for your experiment. Not only the amount of cells, but also how much RNA or DNA have you used for the experiment, for you set up the libraries. Coming to one of the earlier questions, so quality control. So we said we don't recommend quality limits, but we would like to know, and how many reads pass the quality controls on the sequencing machine. So this built-in quality control set up with the manufacturer and the version number of the sequencer. So the sequencing platform here because also software changed and then maybe the output changed. And if you have done any modification of the built-in first quality control steps. Also everything what leads to point 4 the FASTQ files. And so what is the read length, or is this, if there's not specified, it's not Illumina 250 times 250 or something, or is this more Oxford Nanopore or PacBio, there's a different read length all the time. So it's the average read length. Where's the sequence? Is this a company sequence, or is this a core facility or in-house? Batch numbers, when is this run, and which sequencing kit is used. And then this was quite fast now. Core elements 4, 5, and 6. As said, the 4 and 6, actually 5 format. The 4 and 6, 5 is how to process them. So everything before 4, or including 4, can't be redone. So this is 4 is the raw data dumped in SRA or dbGaP or somewhere. Everything after can be modified, remodified, can be redone with different cutoffs and everything. But we still would like to cover some of these steps after creating the raw reads. This is the basic processing of the reads, so aligning forward to reverse read. If you use specific tools, scripts, so which version is it, what have you used, and which quality thresholds have you applied to them, or primer cutoffs, collapsing methods, and so on. And then also free text, how you did it in detail a little bit more, but it's more than the best case scenario is that you really provide the script. Some people may not do it, but at least we want to have an idea with all the quality cutoffs how you did it. And the end product would be then the FASTA or FASTQ file, which can be sent to a VDJ aligner, IGBlast request or VDJ server or something, to really assign the VDJ elements to it and further on in the process. Short overview, we looked in different databases, just example. All the terms we defined already existing in different databases. Not all of them, but a lot of them already exist. And most of the time they have much more annotation to it. So you can really pin down in much more detail, but we said, okay, these are the information we would have, at least we would like to have this information. And a lot of database covers them already. And one goal for the next step, or already here, is to maybe homogenize some of the database that they have a standard agreement on. Okay, we all cover this minimum set of information in a similar way that they can be maybe later on comparable, but I think the database group will also say something about their ideas for the future. But the really idea is so that everyone agrees on the minimum set of terms. This can be always expanded, can be always changed, but first is we have to start somewhere, and this is, I think, the right time to do it. The goal was last year to define this minimal standards or minimal information with data submission, and our plan is to talk about these terms, get a feedback, and if we can, we would like to move on with it if we have an agreement on that, and make them public and try to get an agreement from different sides on it. The agreement should also include companies to the question, so we would like to have companies on board. We will figure out which term, or companies one thing, kits is other thing, that you actually do the sequencing yourself, but you're using a kit for doing it, but then you have this information. There's checklist including the terms, what should we change or exclude, included, expanded. Then, as I said, we will release a draft of them so everyone can have a look and give their comments, either online or in person to one of the members of the working group. And we have time for discussions.
Right. So it's just amazing work. I'm amazed at how thoughtful everything is. I had a question about core element 1. I don't know if you can bring up that slide. So I just happen to have come, so I'm a pathologist, a practicing pathologist, and I just happen to come from a meeting where we were discussing these kinds of things on the kind of pathology informatics side for the College of American Pathologists, our big national thing. And so I'm a little bit PTSD about this. But when I look and I see, I guess it was the next slide about study, or the third one about diagnosis. There we go. I'm sorry. There we are. So was there much discussion about the kind of nomenclature or standardization for things for entries under, say, diagnosis or intervention? And thinking about, I mean, there exist these kind of rather terrible but existing codes like ICD codes and CPT codes for those things. What were the committee's thoughts about that?
Well, yeah, the ICD-9 and now more recently ICD-10 codes. One of the challenges with using those is that they change a lot. What we're hoping to do actually is team up with existing databases, and in particular the IEDD. I think would be the DB would be the one that we would want to perhaps target for assistance with this, because they have a pretty advanced setup. But Steve, you wanted to say more?
Yeah, no, that's exactly what I was going to say. So Bjorn Peters is here, and people may know the IEDB database, which has captured sort of antigen-specific sequences for over a decade now. So they have a lot of experience with sort of capturing this level of information for these kinds of data. And so we've talked to Bjorn and the team over there, and we've been borrowing and trying to from their experiences in coming up with this. So at a first pass, we've not gotten to the level of controlled vocabularies or mapping these terms to ontologies or any of that, where we're sort of just listing out the kinds of things that we want, the kinds of information we want to capture. We do envision, once we can sort of take this step, that in the next stage we're going to want to try and get more details on how these fields should be named and how to map them to ontologies and what are the terms that we're going to allow or not allow. And that, as we get to more of the implementation details in the databases, that's going to become important.
But I think in terms of things like ICD-10 codes, it can get to be too much. So it's very challenging to, there's not just the disease entity that some of those codes capture, but also other accompanying issues, and it can be very challenging to code things even in real time with those codes. Speaking as a pathologist myself also.
Yeah, so I'm not proposing that one use those, but thanks for how you guys have thought about it.
I'm a bit confused about the minimum standards for publication versus the minimum standards for data deposition. And if you think about minimum standards for publication, I don't quite see what we're asking a journal to do here, for example. Do they, when you submit, are you supposed to fill in this detail somehow through the journal portal? That probably wouldn't work. So what you had in mind? Or?
What we had in mind, so if you look at, for example, what's required when you publish microarray data or RNA-seq data or other high throughput data types, when you are going to publish a paper that uses those kinds of data, the journal will ask that you give them a study identifier from a relevant repository. So if you are going to publish microarray data, they will ask that you give them a GeoID to show that that data is deposited in a database. Or for RNA-seq data, you might have an SRA ID that will be required when you publish the paper. We would ask for the same thing. We envision that there are going to be data repositories for this kind of data, whether it's SRA or some other repository we can talk about, and that when you publish the paper, you should be required to submit an identifier that shows that you have deposited this kind of information along with the data in a quote-unquote approved repository.
Therefore, for the journal, the requirement is that the data be deposited in a database that adheres to this standard rather than any further standard for the journal.
Thank you for your work. I'm totally on board with your, like the requirements, the minimal standards. Both the raw FASTQ and the processed FASTA FASTQ files, I think, are definitely required. My question is about step 5, the actual processing. And Florian mentioned it would be nice if people upload. Why not seize the opportunity and really require people to upload their scripts? Because I think like a free text description of how the algorithm works is a really bad substitute for the actual algorithm. And then I think building on this is my question, is there any precedent where a data standard has forced proprietary companies to open up their black box? Has that ever happened? So yeah, I don't know. So I guess that was 2 questions. But
I don't know. To the last question, I don't know. And I don't know if you have right now the power to enforce it, to open up the black box. I mean, what can we do if they say no? As long as these people still buy the kits and run the sequences, and as long as the journals, I mean, if journals say you have to release all the data, then maybe, yeah, otherwise it gets complicated. But this is actually the last point with the free text or something, is exactly the problem. So the companies don't release this step 5. This is hidden most of the time. Yeah, I would like to put some pressure on it. I'm not sure.
This is a really good question, and we had a very animated discussion on this, I would say, before lunch, and I'd love to continue it. I guess my personal opinion, and I guess what we came to as a working group, in an ideal world, I'd love to capture the code and all of the steps that were used to get from the raw data in 4 to the processed data in 6. I think it would be great. I think right now it's a very, very difficult thing to do. It's a very complex thing to capture, and I don't think that the overhead of doing that is worth what we would gain as a community from capturing that particular step of going from 4 to 6. I do think there's a lot more interesting stuff going on after 6, from the whole VDJ assignment and clone definitions and things like that. But this is one of the things we should discuss. But our opinion, the thing we're recommending right now as a starting point for discussion is that you would not have to deposit, an exact code or virtual environment or anything like that that would explicitly take you from 4 to 6.
We would like to have it, but it's maybe too far for now. This is, yeah.
Thank you.
So just to point out, perhaps it's obvious, but a script isn't necessarily a good substitute for a description. And I actually, whether the code works or doesn't work is obviously important, but I think listing the key points that you're aiming to achieve in QC in a form that everyone can understand is more important still.
Yeah. This is a point for a little more detail. So first, really amazing work, and I know how difficult it is to even get to that level of trying to get to a standard. First, regarding, since this slide is here, I think that even if you just state which company pipeline you used or which software you used or whatever, for people who afterwards want to assess it, it's already something. I mean, it's better than nothing. So I think that even is a big thing. I have 2 points. Somebody mentioned quality before, and I understand that requiring a certain quality standard is totally different question than what we want to do here. The question is, do you want also to require that the quality is going to be reported? And you mentioned quality regarding the sequencing, but I haven't seen anything regarding quality of the samples, either in the cell level or in the nucleic acid level. What about reporting cell quality or RNA quality, anything on that level. That I think is a very interesting thing that many times is the problem when you're assessing the data afterwards, if you want to reanalyze it or just assess it or anything.
Yeah, good point. And to the companies, yeah, one idea is that release at least a version number.
Yeah.
So this, you don't know the pipeline, but this version 1.2 instead of version 1.4 or something.
And the other question, if you can go back to the sequencing and to the barcodes and all that, I think it would be worth to be a little more detailed here. And in most reports, you get both from companies and from different pipelines I worked with. You do get already more detail than that, even in standard reports, and I would require more detail. And what do I mean? I mean actual assignment of barcodes to samples, so actually require that the barcodes sample mapping is going to be included. So in almost all reports I get, there's always the mapping of the barcodes and which samples. This is very important. We have found out that barcode impurity is a very big issue, and if you're able to assess which samples which were sequenced with which barcode, that would be important. And also the lanes and which samples were in which lanes or in which batches, etc., etc. So I'm not sure if you want to have it here, but I think it would be worthwhile to require it in detail, that the mapping of the lane, the batch, the flow, whatever, in which date each sample was made, because many times people are going to give you all the samples of the study in one big file, and it's not enough just to put one date. You have to really give dates and everything for each sample alone. I think that would be very important.
Yeah, completely agree. Good ideas. And the general principle was that data submitter should provide sufficient information to go from the raw data to that process data, which would include things like those mappings. I mean, we haven't explicitly split that out into multiple different data elements, but I think, you know, it may be covered under this sort of element of the description of how the barcodes and the primers are organized.
Yeah.
Yeah, look, I agree that I'm amazed how much you've thought of, but can we go back to core element 1? Because no, that'll do. No, maybe the next. Yeah. Oh, okay. No, maybe you've got it. And I'm foreshadowing here something I was going to raise tomorrow. Are we here? Of course, I think everyone here probably is interested in the human repertoire. But I think what we do here should extend through to all species. So animal, human, or synthetic might be species name.
The species names.
So a continuation of the previous question is that in terms of the germline sequences, any plan to also monitor the germline sequences? Because at the end, the accuracy and comprehensiveness of the data is going to be coming up from that. So is any interaction with the genomic sequencing community, or also with the IMGT, for example?
My understanding is that the tools group is going to have a very detailed discussion of that.
Yeah, they will cover it. So they cover exactly this topic.
So I'm thinking.
So let me just say so we have an online shared document, a spreadsheet that includes all of these different data elements along with descriptions, mapping from our recommendations into IEDB, EJ Server, all these other databases, which we will make available for comment, and we would absolutely love to hear if you think there are certain elements that are missing, redundant, not necessary, that you should comment on it and let us know. this is what we're hoping for.
Also, please, if you do make comments in the document, could you add your name and email address so that we can follow up with you if we have any questions about what you're asking or commenting on?
That was a great start. And those of us who have done these meetings before, they can be exhausting. So I think maybe we'd take a 15-minute break, and the coffee shop is still open, and then we'll come back and do Common Repository.
I don't know. I don't know if you see it.
Yeah, that's us. Yeah. Okay. So a couple details. I'm going to go online and order my box lunch with my non-U.S. Credit card. I was told that I can do that. And Jasmine mentioned if anybody has their talks in their, on a key already, they could bring them back to Jasmine for tomorrow. That would always keep things going. So as soon as you have your talk, you have it now, you can give it to Jasmine. I think it'd be a great idea if for the first few times people ask a question or make a comment, maybe give us your name, because I've only like going, I know that person, but only kind of. So, and we've got 2 more groups that have done an incredible amount of work. So now we'll turn it over to the Common Repository Working Group.
Hello, I'm Yarif Wine from Tel Aviv University. So Common Repository Working Group consists of all these people, and we were e-meeting over the past 12 months. Twice a month through Skype to discuss issues and ideas around the development of a community-wide common data repository. All the recommendations, are outlined in a recommendation draft, and we identified 4 key areas that I will detail later on, and 2 levels of compliances. So it's a bit complex, all the recommendations, so what we try to do is to put it or lay it out in a graphical overview, and as you can see here on the lower part of the slide, we have the administrative recommendations and the technical recommendations, and the focus here are on required recommendations, and the numbers appear in the document. And you can see here distributed repositories under the umbrella of a central registry. So this is the two level of compliances and the lower part. And the upper part emphasizes the required recommendation in the four key subjects we talked about, or we put together: data sharing and access, infrastructure, data to be shared, and NIH coordination. So what I will do, I will go through the key recommendations of each section, and Yuri will continue on the infrastructure and NIH coordination. So overview of data sharing and access. We dealt with data sharing requirements, mandates, access restrictions, use of protected health information, donor informed consent, and IP issues. And here again, in order to understand better which recommendations are subjected to which recommendations, we have here the graphical overview of the detailed recommendation for data sharing and access. And the ones that are highlighted are the required recommendations. And I won't go through all the other recommendations, but you can see the highlight of the recommendations in these boxes. So recommendation number 2, open access, and this is a very important recommendation. We strongly recommend that all repository or distributed repositories will be provide open access to all the sequencing data for redistribution and reuse. Anyone can freely access. You don't have to log in to access this data, and you can use it for any purpose. And this in the position and line with other all the trend of science towards open data. Now, we recognize other open access database without restrictions as HLA typing data, RNA-seq data, cancer sequencing data, which has no restrictions. The second recommendation, or the required recommendation under data sharing and access, in line with recommendation number 2, is regarding intellectual property. So once you submit the data, you have to be aware that this data is not protected under intellectual properties restrictions, and it can be used. If you don't want people to see it, just don't deposit it. So these are recommendations regarding data access and sharing. What will be the data to be shared? So the key points are the deposition process and raw data in registered repository and existing database. Here again, we lay out the recommendation in a graphical view. And as you can see here as well, we emphasize recommendation number 14 regarding raw data, QC data, and annotated data. And actually, in a meeting with minimal standard group, we recognize already a gap between the endpoint of minimal standard group and our recommendation, something that we will need to address further on, especially this point, the annotated data. And another recommendation which we have requirement for be included in our repository is a link to raw data in SRA. So we strongly recommend sharing raw data sequence and quality score data, and as well processed and annotated data. We we know that many companies produce or generate a large amount of data today, like as you see here, these the names of some of the companies. And we feel that it's important to engage these companies in order to find a way for them to generate data or prepare the data to be compatible with the repositories in our recommendation. So this is an important point. I'm not sure it will work. And recommendation number 17, the link between the raw data or the link between our data to the raw data in the SRA, if it's annotated data or any other data, as you can see here in the in recommendation 17. So we need other, or you can see other recommendation related to the link between the deposited data to the raw data in SRA. Okay, this I'll leave for Yuri. You have any questions before I?
So as Yuriv mentioned, we're trying to define what a repository looks like, and we've identified that it kind of splits into two different camps. There's administrative compliance for what a repository should look like, and that relates a lot to kind of policies around sharing, et cetera. And the other set of things is really around the kind of technical compliance for what a repository should look like. And really this is, in large part, what we're talking about is machine interoperability and how to ensure that multiple repositories can speak the same language, how all the tools we develop can speak the same language, et cetera. So from a high level, the main infrastructure that we're kind of proposing is to have a kind of distributed set of repositories, all of which would comply with the recommendations or with some subset of recommendations that we suggest here. And in addition to that, there will be a kind of central registry that can describe to someone these are all of the compliant repositories that we are aware of, and some of them can be application specific, for example. And we're also not precluding the possibility that we would endorse creating a kind of official AIRR central repository as well. I think that's a little bit up for discussion. And so let me just go straight to the recommendations. So first of all, we're really looking at a distributed approach. There's obviously advantages and disadvantages for doing distributed versus purely centralized. Some of the advantages of doing distributed are the fact that you get, there already are multiple repositories, and so it'll be very hard to force everyone into a single centralized location. And in some sense, having multiple repositories lets different ones compete to see who is offering the kind of most useful services. Agreeing on standards for having repository interoperability is something that is in some ways very much a solved problem, so that's not really an issue. And multiple technologies exist for doing this. There's already kind of somewhat of an analog in a similar effort called the Global Alliance for doing human genetics as opposed to repertoire sequencing. Obviously, disadvantages of doing distributed repositories include the fact that we may be duplicating effort. It's not necessarily obvious how we're going to deal with synchronizing data, or if data gets deposited in multiple places, and whether we want to actually support some kind of federated queries across all of the repositories. But overall, I think that the committee is making the recommendation that distributed approach would be advantageous, at least for now. So in addition to having a repository that actually contains the data as described by the previous working group, we also want to have a central registry that would list all of the potential repositories that are out there. And in order to be listed, you would imagine that you would need to, for sure, achieve technical compliance so that we know that all the data types are the same and everyone's tools that are expecting to use this data can all access to all of these different repositories. And in addition, we're recommending that in order to be listed, you should also adhere to the kind of administrative compliance recommendations as well. So ensure that the data is kind of open access and has the same kind of like IP guarantees, et cetera. In addition, we're also kind of thinking, we haven't really like fleshed this out in huge detail, but thinking what are the kind of queries that the different repositories should be able to support. And so in some sense, you can imagine that having a repository of immune sequencing data would present to you a set of different things you can ask for. So you can say, I want, give me all the studies that match a particular set of cell types or subject types, or even in finer granularity, give me all the receptors that match a particular set of annotations. But these things still need to be fleshed out a little bit more. We also strongly support using kind of a standard open source data serialization format instead of kind of coming up with our own custom file formats. And so this is the kind of tool that's common in kind of common in industry in kind of big data industries where they use tools like Avro Thrift or protocol buffers, which all basically come from somehow trace their lineage to Google, where essentially it's one of the ways that we recommend to kind of implement this machine interoperability. And so by essentially specifying the schemas that were presented in the last hour, using one of these tools, you can essentially get access to multiple file formats. You can ensure that multiple programming languages can all query the same databases and that kind of thing, and everything kind of comes for free. And I can kind of go into that in more detail later on as well. But currently we're recommending that we use one of these and potentially Thrift. But this is potentially controversial, so if there are any kind of opinions on this, we would love to hear them. As I mentioned, we recommend that repositories implement a defined set of queries that people should be able to run on the databases. And we have not proposed a specific set of queries yet, but hopefully we can dig into that a lot here. And then finally, hopefully a lot of these standards are kind of consistent with NIH Data Commons that's kind of being funded through like the BD2K efforts. Yep. So to repeat what I just said, we recommend being compliant with kind of community standards for ensuring kind of data interoperability and database interoperability. So with that, we are also happy to open into any questions. We're looking for any kind of community feedback on kind of what kind of queries you want, whether this architecture is going to be problematic for anybody, etc. Oh, yeah, all the way at the beginning? Yeah. So in addition, we put our recommendations on a GitHub repo, and we would love to kind of engage with the community. Feel free to submit issues or even pull requests if you have kind of proposed changes. And then hopefully, we can be up for discussion, but hopefully if this is successful, maybe we could even consider putting additional recommendations on here as well. Perfect job.
If you would like to have buy-in from industry, did you consider the whole open source and no IP aspect? And how do you weigh that in the whole discussion? Well, just, oh, sorry, my name is Rick Rademaker. I'm from GenMap in the Netherlands. So basically, like Felix already introduced, getting industry involved in this project is well, at least a discussion point whether that's favorable or not. But if you would like to have industry buy-in, what kind of discussions did you have, and what kind of considerations did you have in order to make the statements that you want to have a completely open source and no IP protection as a recommendation?
I don't believe we've had any specific discussion with industry. So to us, the position of this data would be analogous to the kind of data that is submitted to, say, GEO or SRA for other things like RNA-seq or other analysis. I do not. Essentially, the kind of data that ends up in the repositories would be what would be currently in supplemental material to papers and stuff like that. So I don't think that there should be any insurmountable IP hurdles specific to this subject. I think it's just that the area is new, and as always, there's pioneering industry people out there, and we are hoping that this will essentially solve itself. That might be overly optimistic, but I would be surprised if there's this is a very competitive space. If there's going to be some industrial partners who are willing to submit data in a way that it actually helps the whole community work forward, and others that are not, I think it'll settle itself rather quickly which industrial partners would survive. Also, hopefully funding by NIH could be asking for actually maximum value of data sets that are generated based on taxpayers' money to not hold back key information that is necessary to get maximum value out of the data.
Also, from a technical perspective, the kind of distributed design makes it possible for industry to buy into the data formats at least, so that they can also participate in kind of a machine interoperable sense and still kind of run their own internal repositories. You can imagine that maybe in the future down the line they could find reason why it would make sense to open some of them or provide limited access. But the idea is that technically we make it easy for everyone to buy in and then to be kind of listed in the central registry. You should be willing to provide kind of unhindered access to the data.
And also, there would be no IP on data in the database that could restrict others' access to it, but similarly, there would be no restrictions preventing industry from accessing and using the data that's in there.
Yeah. I think the point of open access, I mean, from a scientific point, it's a very relevant one. I think it will be hard to defend in a commercial perspective. I think people from legal will and IP will, within most commercial companies, having problems with such a model. I think in terms of formats, I think open formats is what you also recommend. I think that I would be a strong support for having standardized open formats and not proprietary formats or company-specific ones. So I think that's a good one. That's a good option to aim for. I think also last year we had discussions about personal data, and we had discussions about DB gap and protection layers, and that is something I also don't find back in this discussion. So are you allowed to put patient material that can actually be traceable back to the individual? Can you put it in an open database? I don't know if there's anything, if on the regulation level, if there's anything that has been changed so far. I think last year we had some discussions about that, and it kind of conflicts with what I hear right now. So do you have some opinions on that?
Right, that was this principle. The recommendation number 2 was meant to address that, and we were of the opinion, and also consulting with some experts in the field, that really the risk of re-identifying data from raw RNA-seq data that is deposited would be just as high as from repertoire sequencing data. And raw RNA sequencing data is routinely reposited freely without any attempts beyond the regular de-identification and removing any kind of health record data from this. Similarly, HLA typing data, if you make that available alongside with any kind of other information about, say, age or something, would in principle allow you to identify an individual rather easily. So I think this is the area. If you're really serious about there shouldn't be any information made public that allows you to identify a person, you should actually stop publishing, say, in Journal of Immunology papers about, in this hospital I had a child that had this rare form of cancer. At that point, you can already identify that child rather clearly. So in our mind, we are not going beyond what other people are doing routinely in other fields by asking this. And the general discussion of what re-identifiability means, I think that's going to be a general area that's going to be in all of science, going to be really important to clarify further, and there's clearly a gray area there. But if we're comparing ourselves like clearly whole genome sequencing data currently ends up in dbGaP, and that seems to be at one extreme, and that's useful. And when we're comparing ourselves to other things that are currently not ending up in the protected repositories like HLA sequencing data, RNA sequencing data, or somatic mutations in cancer, then we feel like repertoire sequencing data is not more dangerous than those.
And also we had a supporting recommendation that specifically addresses personally identifying information that just states that any repository which chooses to host personally identifying information do so in such a way that the securing of that data doesn't impede access to the repertoire sequencing. Oh, it's my, sorry. We have a supporting recommendation that addresses personally identifying information specifically. And it just states that we recommend that any repository that chooses to host such information secure it in a way that doesn't impede access to the repertoire sequencing data and its associated non-identifying information. So obviously, if your database chooses to host, birth dates and zip codes, that needs to be protected, but it should be done in such a way that that doesn't impede access to the repertoire sequencing data.
So I would like to discuss a little more about where the line is between sort of the central repositories and these distributed repositories that you're talking about. I mean, there was one recommendation that the raw data go in a central repository like SRA. On the other hand, there's a proposal for these distributed repositories. I mean, I think it's important that when the data get published in the kinds of depositions we were talking about in minimal standards, I think it would be helpful if there was sort of a single place that we as a community could recommend that all of the data really ought to go into SRA or a place like this, just like the microarray community has GEO or the RNA-seq has things like SRA, to have one place where all of the data can be stored so we know where we can find these kinds of data sets. I want to know where that line is and how you envision that working.
Well, I mean, I think this is where we start overlapping with you guys, as you know. But man, we're pushing for SRA for raw data. So the question that I think we would like to open to everyone is, raw data applies to year number 4, I believe, on your last set of recommendations. And the questions will be, what ends up in these distributed repositories? What did those data look like? And we don't have the answer to that, and I think that's something that we should all be discussing today and tomorrow and Wednesday. But it all stems from also like what kinds of queries we want to ask. I mean, I think there are a lot of open questions, but definitely we recommend that the raw data be in SRA, sort of forever. But in terms of the repositories, I think that's an outstanding question.
So just as an example, given that the query interoperability would allow anybody to say, okay, I'm going to set up the central repository which has everything. And they can go around and query every single one of the other repositories, take that data, because there's no restriction on it, and say this is the central place where everything is. But someone else may say, my repository, I only want to have quality data to a certain extent. I'm not going to take data that has these and these characteristics. Or somebody may say, I'm going to have just the cancer repository database, and I'm only going to use this data. Or I'm not going to allow any mouse data because I think mice are the evilest things ever. And if from a bioinformatics standpoint you're interested in the repository that gives you everything, I'm sure somebody's going to set that up.
Thanks to the group. A quick question about the intellectual property clause on the data sharing. I can imagine many ways that this could be in conflict with what's normally done. Just a quick example from HIV, a lot of people discover broadly neutralizing antibodies and they patent those antibodies and then they sequence more. And so there are antibodies in that data that are intellectual property, but they're also of high scientific value. So I was curious, yeah, about the intellectual property clause and where you see that going.
Our understanding, there were a couple of attorneys and ethicists on the group. None of them are here right now unless Tanya's here in the room. Maybe she should respond to this. Is Tanya here?
Tanya, I see you. Sorry.
Yeah, so I mean, I think there's different ways around this. So there's different communities have structured this in different ways. So in terms of the industry involvement question, it's a difference between whether you're accepting of the fact that industry may not be a depositor but could be a user. So that's something that you need to come to grips with as a community. This sort of issue about who are the free riders on your community. If you are really opposed to the free rider issue and it's really going to stick in your throat and it's going to become an impediment to data deposit for the community, then you're going to have to put in some kind of controlled access to your database. Where part of what you are looking at is if you want to use the data, you also have to have some form of agreement that you're going to recontribute or contribute that data. So that's a philosophical question that as a community you need to come to grips with. There are experiences, and I was actually going to talk about one of them in my talk on Wednesday. You know, there are examples of open access communities where industry has come to play that really did start within the data space and have since moved right through into probe development. And they have become public-private partnerships, and industry really has stepped up to the plate and opened up and actually contributed to the development of those probes. And there I'm talking about with some of the protein kinases and other cancer proteins of the Structural Genomics Consortium. So that's kind of a philosophical question. In terms of you know, where you want to actually set up your level of protections about how far out do you go with your reach-through clauses in terms of the IP. I think the very simple statement is that if you want to use the data from here, you need to put into an agreement, regardless of what you then go on to develop as your derivative products, that you are not going to you're not going to enforce any form of intellectual property as against other individuals' use of the data in the databases. So this is the way that you kind of contractually structure the commons. The problem is that there's always a trade-off between the complexity in the way that you structure the legal terms, so the permissions and the controls, with the stickiness of the data in the system, as in terms of how easy it is to take out and to put back. So I think you're starting from the right point, which is basically what's the objective, where are we trying to get to? And then once you've kind of sorted out where are you trying to get to, then I think there's different ways to get there, and the way that you want to select is the one that causes the least number of impediments and clauses and least level of complexity is what I'm trying to get at, I guess. Does that answer any questions, or do you want me to be more specific, or?
So just to kind of clarify that, so you're suggesting that the IP restrictions apply only to, say, analysis of the data rather than something like drug.
There is no IP in the data itself, right? So in the data that you're putting in, the data itself, right? Not the antibodies that you're generating or that you're using or the actual sort of substantive material, right? There's definitely, if you're developing antibodies, then of course you can patent those antibodies with some strictures, because there's some confusing jurisprudence, at least in the United States, about whether you can in fact patent things that are structurally the same as naturally occurring materials, right? But in terms of the data itself, generally the data itself is held, the reason that you can't access it is that it's held in trade secret or behind a firewall, right? Yeah.
I don't know. I'm not a lawyer.
Yeah.
So.
Yeah, so the data itself can be supportive of the patent application of the substance or the process or the method, the things that you can actually patent. The way that industry keeps its data, it's in trade secrecy and it's kept behind a firewall. The fact that you can't access the data that is the proprietary element. It's not a patent over the data or even copyright over the data. Yeah. There's no formal IP in the data, except at weird things in the European Union where you have IP specifically over the structure of the entire database. You have database protections in Europe, which don't exist in the rest of the world. But there's no IP in data.
So our main point was, I think, simpler. Okay, in this, 1 second. That if somebody wants to submit data to the repositories, they cannot say you're only allowed to use anything you make this data is going to be owned by me because I submitted this data. That's the main point we're trying to make, and I think we have to work on language maybe to make this more clear. But
my use of the data doesn't preclude anybody else's use of the data in the database.
Can I start? Just wanted to note an intellectual property case that's come up in the case of flu sequences, which is sort of basically the third world countries have said, look, if you want to take all our sequences, which we do because that's where flu starts, and use those sequences to build drugs, we have a bit of an issue with your saying those sequences are free and not worth anything, and then you sell the drugs back to us. And I just think, I mean, again, looking at the IP stuff from a different angle, we say today that there isn't anything identifying in a repertoire and that there isn't anything too terrifying in a repertoire. But I mean, if we wind the clock forward and think about some of the things we'd like to do, you know, maybe in 15 or 20 years' time, we could pull out somebody's entire immunological history from a repertoire. I'd love to be able to do that. I know others here would as well. I just think we need to be a bit careful about these things and a bit open about the limits of our recommendations. You know, I agree with you that it's better to start from a position of being too open than too closed. And from the point of view of scientific research, you know, this is big data, and what we need is a lot of it so that we can analyze it properly. But I think in the way that we say these things, we should shape it as you said, alluding to RepSeq and other things. You know, we don't want to be out of line with them. We want things to move together. But I think in doing that, we should acknowledge some of these wider societal issues that in the end are going to need quite a bit of communication with patient groups, the public, you know, a lot of people around the world. I think we need to acknowledge some of those difficulties.
You're referring to the Nagoya Protocol and basically a whole bunch of national implementations of the Nagoya Protocol, which is around access and benefit sharing. It is definitely an issue in other regions of the world, and you saw that with Indonesia and pushback in Southeast Asia with the influenza vaccines. The issue there is generally around there's a pushback against the open access data movement because there are inequities in ability to use that data. And so, you know, they can be benefited in different ways, usually through scientific capacity and or through agreements if there are and products that are developed. I agree with you that those global tensions are really becoming quite real as between, you know, developed and lesser developed countries. And they play out in the patient realm or in the patient world. We were just talking about it with Jamie before, where you have a tension between the two ends of the spectrum, where you have free my data movements of please have at it and find cures for my rare disease, as against working with vulnerable populations, particularly with indigenous populations that have interesting genetic profiles. And so in that case, having those levels of different kinds of security over some aspects of the data so that it's not completely open, but there are corners of the data that need to be protected for societal and ethical reason. I think it's important to keep that in the back of the mind. But it's not the 99% of the database. It'll be a small portion of the protected database that has specific strictures attached.
I agree with everything you said, right up until it doesn't apply to 99% of the data.
It might be 99, might be overstating it, but I think that the bulk of the data you can keep as open. But I think that there are things where there's traditional knowledge attached, indigenous or vulnerable or specific populations attached, developing country issues. That certainly applies with some of the communities that I work with in conservation biology. Or whether you need to be able to identify those and carve them off. But I still think they're an exception and not the rule, and the rule that you want is as open as possible, as much as possible, whenever possible.
I think my point's really around sensitivity to issues that might develop, and those issues might not even be real. I mean, in the UK, we've had a lot of pushback against the idea of medical records becoming open to companies. And that's, to my mind, largely about suspicions that companies will do things with them that they probably won't do. But I think it's very important that we approach this in the right way and we show sensitivity to these issues. And we perhaps don't presume too much up front. This is sensitive and that isn't. In the end, that's going to have to be a consultation with, not sure who, but a much, much wider group of people than just us.
I mean, I think ultimately that also comes down to what we think we want to achieve with this repository. The second you start talking about dbGaP and those types of things, I think everything changes. The stuff that we've recommended just doesn't really mesh with those types of things. You can't just walk on and use those data as you're discussing. So I think that I agree, and I'm sure everyone here does agree that we need to be sensitive about these things. And if that's the way we want to go, then we need to decide that as a group. But it's not where we've been coming from for the past few months. I think it is where we started, but it's not where we've been coming from lately. And I think it does change everything. It flips everything on its head, actually, because I just don't think you can approach these problems in the same way if that's what your major concern is or your primary concern. I don't know if you guys have anything to add to that, but
I was going to say this person is
Yeah,
really patient.
Yeah, I'm Jim Crow at Vanderbilt. So I wanted to comment on the idea that sequences are not IP because I don't find that to be true. So if for the people, and it's not only commercial people, for the people who are doing antibody discovery for particular targets, of which antibody are now 40% of the drugs in the pharmaceutical pipeline, these are major classes of drugs. There's a current, I mean, you alluded to the... Well, because of Myriad and other recent decisions, it is not clear that you can protect sequences as intellectual property by patent protection, and therefore many groups are keeping the sequences proprietary in order to own them, and the U.S. government nor universities manufacture and develop antibodies. They give them to companies, and companies only pick them up if they have exclusive ownership usually. And currently, a lot of that is done by proprietary sequences, which might be transferred from academics to a company. And if the highest principle is not open access, the highest principle is supporting public health. And if we have antibodies and we cannot protect them and therefore can't give them to companies, and companies don't develop them, then the public health is harmed by that. So I think, And we also have law by DOL, which compels us through our federal funding as academics to protect that IP to therefore long-term benefit public health. So, you know, by DOL supersedes regulations about open access data, and I think we have to work through that we have to allow sequences to be protected, even if they can't be protected by patent, as property that could be given to companies to support the product development. And if we just categorically undermine that, then we've lost. You know, we've lost the ability to develop those antibodies and invest in them. So I'm on board with open access. I like the idea, but we have to recognize some of these sequences are valuable, and they, you know, if we compel federally funded people to disclose all sequences no matter what, then, you know, that contradicts law, basically. And I don't think it's sustainable.
So I'm not the lawyer at all, but so and Jim, we've worked on this for quite a while on the epitope side of things, right? Which is kind of the corollary. So there typically, once we have something that might have some IP value, there's a invention disclosure which is internal, and then we carry on forward with the epitopes to actually generate functional data that there is actually something useful with them happening. And I presume this is the very same as happening in case of the Repertoire data. You start off with the Repertoire sequencing, and then you express the antibody and do further studies with it. Just the fact that you have a sequence would never would never give you a patent or IP protection, right? I mean, it might be that's the first step, and you disclose it and you carry it forward. And I don't think we are necessarily saying you have to submit. I could totally see how you would not disclose some of the things that you sequence and just submit the stuff that's not. But what we want to avoid is that say you've run a query on the repository and you retrieved a sequence that somebody else has generated, and somebody else may have generated the same sequence you have. Does that now invalidate your work on the sequence? But what we're trying to have is working on the repository should not suddenly make you subject to someone else's IP on just querying that repository and finding a sequence in there. Right? If a company says we are depositing here our patented sequences in there, and you're saying the moment you want to develop that as an antibody you want to take forward, you're going to run into their existing IP, which is going to block you from doing that. But what we want to avoid is that you're by using the repository as a user somehow are subject to IP of someone else who's been doing that when you do some other work and not trying to patent that thing that someone else has already carried forward. And I'm sure we have to have better language of spelling this out.
Also, is that does that describe a situation as it actually occurs in reality, in the sense that if you discover some new potential useful antibody sequences, would you not take moves to protect them from an intellectual property perspective before you would deposit anything anyway? And so once it would be deposited, it would already have gone through the kind of legal mechanisms to protect them? So Jim was saying that after the Myriad case, it is not possible to patent naturally occurring sequences.
I agree with a lot of the stuff that has been said, and I don't think it's possible to get IP on a whole panel of antibodies. It's definitely something. So once we have identified an antibody that we want to develop as a therapeutic, we're definitely putting the CDRs, for example, in a patent, right? And if that's not possible, then we don't have a patent. I mean, we can develop a compound, but it's very easy to mimic or to regenerate the product. So basically, the value of the product is nothing to us. So that will be hard to develop, I think, in the end. I don't think the aim of this database is to get IP on the database, but to have some sort of protection on the sequences that you provide over there. And if you want to develop, or if you want to maybe not even develop a compound, but actually want to have a compound that you want to use for testing purposes or for feasibility studies or whatever, you need to have some sort of access, but also need to have some sort of protection on these antibodies so that you can use it and that you can share it with the rest of the community. I think if there's no sense of protection at all, then it will be very hard for companies to actually provide their sequence data in these repositories. But I'm not a lawyer, and I don't speak for my company. This is my ideas about the subject.
I think beyond the IP, it's important to state that the repository is a platform to accumulate a lot of data. You can, as a community, we can learn a lot from the data deposited there. And I think also companies can access the data and not look for that gold antibody, but more learn about trends and more learn about processes, development, immunology. And this is really important as a community to push it forward, not thinking all the time about how to protect. We want to open to progress research in these directions.
Okay, so basically it comes down to the question whether you want to have commercial buy-in, so companies that actually contribute to the database, or that you allow commercial companies to use the sequences. That's the case what you're saying, so companies are allowed to use the sequence database and that's it. Of course, but then you don't have buy-in from the commercial partners, right? You don't have any, probably won't have any companies that will contribute to the database or actually pay for access, or maybe even pay for access. But, these are points to think about when you're setting up such a platform.
Yeah, and I think that's exactly what Tanya said. That may just be life, and we have to deal with it. So, yeah, I think it's a really good point. I think that there probably isn't a perfect solution.
Quick question. Any thoughts on how you might convey the argument for open access to IRBs?
Well, we do have as our part of our future work to develop consent language that we would just provide to the community as a service that people could incorporate into their IRB protocols and their informed consent documents. Obviously, the repositories wouldn't be requiring submitters to use that. The language would just be provided to make that easier process for people who do plan to submit. But we haven't yet developed that language.
Just want to make sure that everyone understands that what we're looking for is a set of recommendations at the end of the week, and that this has been an extremely productive conversation about IP, and it seems to me that's something that we're really going to have to work out. So as it's going on, I just hope that people will recognize that what we're looking for is going to be something along the lines of what is on the slide right here. And if there are specific ways that it ought to be revised, then let's be shooting for that. Let's think in large terms now, and when we talk about this tomorrow or Tuesday, and then by the time we get to Thursday, it would be absolutely wonderful if we could have something crisp and concise like we have up here on the slide.
Please submit a pull request.
I was just going to say, make another plug for go to GitHub and edit the document and submit.
So actually, and so we should probably preface with that. You are trying to be very concrete, and please also read the whole document rather than just the things here, because it's always easier to criticize something when we are really trying to be explicit of what we prefer. And this is a recommendation of maybe we're 8 people or so. Obviously, we want the broader community input on this, and these are reflecting our personal opinions at this point.
This one very simple piece of language that is from the Beacon Network, and it just basically says, with respect to this IP issue, is that what is prohibited is interference with any other individual's use or enjoyment of the service. So basically, all you're doing is making a commitment to not interfere with other people's ability to extract from the database.
Assuming that it might be time to move on. A completely different question. Do you have any views on how the metadata that the last working group talked about should be linked to your depositories? I mean, again, would you see a central repository of all that metadata, or would that be distributed in some way?
Yes, and we have as future work for our two groups to work together to specify in a computable way those metadata to facilitate submission and query over the database.
But all of the data, everything that was presented by the Data Standards Working Group should be in the repositories.
Right, but I mean, you know, one of the things that's happened most in my mind is I don't think that the SRA has the capability to store the detailed stuff we were talking about in a structured way, and let's assume it never will, because we probably can't influence it. So what do we do about that?
They are willing to do some customization for specific communities, and we do plan to work with them.
Oh, okay.
But you are right, they likely will not in the end accommodate the full level of detail that our community ends up deciding on and will want to have, and so the repositories will be responsible for that.
And then as a short answer, the answer is yes, we have a strong opinion on how to do that, which is to use, for example, Apache Thrift to encode the schemas that were described earlier, and then those can essentially be used to actually store the data in one of basically your favorite database system and made available to people.
So where does VDJML fit in this? Does that question make sense? Maybe you should explain it quickly.
Right. We haven't really discussed that in either working group, so I would say the answer to that question is not known at this point.
We thought that would be dealt with by the data standards people, and so I think this is exactly where we need to coordinate going forward.
Yeah, right. Yeah, it's something that Lindsay started, I guess, mostly. But I mean, I guess I'm just having a hard. So in my mind, VDJML is this markup language that has all the columns that you typically see once you annotate a gene, and I'm just trying to figure out where is all that, because I didn't see it previous one, and now I don't see it again.
It's after number 6 in the previous talk.
I think we were discussing earlier the potential need for also structuring exactly what that should look like. I think some of the issues are that there are many tools to do that last step now, and it's potentially more difficult to come up with a common representation that kind of makes everyone happy. My personal opinion is that it's not, and that we should very much incorporate something like VDJML into our standard, but I think we're going to be discussing that a lot going forward.
I guess to that effect, I mean, the vision that I think we would like to see is that when you publish the data and when you deposit the data, there ought to be some central repository like SRA or dbGaP, which would take the kinds of data that we were proposing in minimal standards, up to the point of where you're going to submit the data for VDJ assignment. VDJML then sort of captures all of that processing about assigning germline VDJ segments, about grouping the sequences into clones, and so forth. And I think that information, I think VDJML is a great way to capture that. And then we envision that information would then be stored in these other repositories that we're talking about, but they would all link back to the central repository like SRA, which would have all this underlying information.
Discuss on creating, that's a gap I was talking about before, discussing creating a standard of what are the actual, let's say, data points to be stored in these repositories to enable queries across distributed repositories. So if one repository doesn't have these pieces of data, the query will fail when querying that specific repository. And if you create a minimal requirements, what data should be there, so then you can actually choose the repositories you want to query from and actually get an answer, not like a blank, not found or something like that. So that's a gap between we have to figure out between our two groups.
Would you have a tool that would search all of the databases simultaneously? As an analogy, you can go to some travel websites and search for an airline ticket, and it will search multiple databases and give you the results. So if we're going to have lots of individual repositories, as a user, I don't want to go to each one and submit my query. I want to do one query once. Is that something that's feasible? That's the idea. So then if I wanted the raw data, that's where I would go to the NIH SRA. But if I wanted the process data and to query CDR3 sequences, for example, I'd go to some meta search engine, right?
Well, it sounds like the schemas that were developed so far would basically have a pointer into SRA somewhere, which is where like the bulk raw data is stored. I think it's always very helpful to kind of think in terms of implementations and kind of what do you actually want to build. And so you can imagine that you want to build a database that you can search through all of the different studies based on all the different metadata. And that's for sure supported, and all of the work that's already been done very much allows for that. I think the question now is whether we want to also support the ability to search actual antibody sequences and T-cell receptors. And that would have to be done there. And it would be very easy to implement such a thing. The other point I should make, by the way, is that in terms of like distributed repositories, it could turn out that it ends up kind of being like a GitHub model in and of itself, where Git is distributed. However, GitHub is basically the central repository for Git. And so you can imagine that there is an AIRR central repository, which will in fact contain most of this data, and that there would also be potential other ones that are very specific or niche.
You may have already addressed this, but I just want to check and make sure. So if one is going to do searches across multiple distributed databases simultaneously for, say, a DNA sequence, is that are there any technical hurdles to that? Or is that something that's actually very straightforward and can be done without any trouble?
I would say both. There exist like great tools to allow that kind of thing to work. I mean, it's not like a trivial thing to do, but there exist very good tools for that.
Non-trivial, but also not impossible either. Okay.
That's one discussion that we all need to have, is that I think what Uri's saying is that these things can be set up, but as a community we need to try to decide and at least land on an initial set of queries that can be supported. Not that those are the only ones that will forever be, but that we can add to the list. But I think if we don't know what we want to search for, then you obviously can't set it up. It can't just be like a wildcard thing. So
yeah, for sure, one of the prerequisites has to be what is it that you want to allow people to search across people's databases.
I also have a question about the distributed databases. Then, what is the level of quality control that you want to have there? And can you guarantee, for example, that data that is now available will be available in 5 years? Or is there a chance that people will go to a different lab and actually part of the repository is then lost? And so how do you deal with that situation?
I would say there's no quality control there, in the sense that you can choose which repositories you want to run your query against. Ideally, so first of all, raw data going to the SRA should basically be forever in principle. And then for these other kinds of repositories, you can imagine this, again, you want to have a repository that provides you certain guarantees, and then you don't want to like legislate to every single other repository exactly what guarantees they're going to provide to people. And so basically, if we have a central repository, we can imagine that we have guarantees that we are all comfortable with as a community.
Yeah, I mean, right now these distributed repositories are obviously being funded by granting agencies that may or may not continue to be funded. So I don't think it's just there isn't a guarantee out there. SRA is the only way we can guarantee that, which is why that's one of our required recommendations, is that the raw data is stored forever.
Yeah,
I mean,
I'm still confused about the motivation for putting all this effort into a distributed infrastructure. I mean, if we put, like I say, we put the base data, the sort of raw and minimally processed data into a repository, it's kind of like the microarray community. I mean, we have Geo, where everyone can access the data, and then third-party groups, bioinformatics groups or other groups will go in, they'll take all the data from Geo, and they'll make interfaces where you can search all the normalized data, you can do all these kinds of queries, and you can go to those resources and use them. The requirement is that this base data is stored somewhere.
Yeah, so the base data is stored, and anyone else can comment on this as well, but I mean, I think that not every group is a bioinformatic group that can go in and download 20 data sets from SRA and do that. So I think these repositories are...
Other groups are going to come in and develop those tools that do sort of take all of the available data and merge it together and integrate and make that available.
And
I don't understand
those would be the distributed repositories.
I guess I don't understand the motivation for making that a distributed repository.
Well, because there are already 5 that exist. Yeah.
Yeah, I mean, so first of all, I don't think it's extra effort to create a distributed depository system. I think it's just as easy. So that.
We're getting the wrap-up signal, it looks like.
Yeah, so it's definitely just as easy to do a distributed repository. I think what I believe is some of the thinking from the group is that given that this is still a relatively new field, you don't necessarily want to make it hard for people to store data and make it available with like the conditions they want, or they want to have, some people want to have their own databases, etc. And so this makes it easy for people to both have their own policies that comply with some minimal set, and also easy for us to have a central repository if we want.
Yeah, but that requires us all agreeing on what that central repository is, which I think is a much harder thing to do. So this allows sort of free flow for the time being. It doesn't mean that we have to say, again, this is forever. This is, right now we know there are 5 repositories that are out there. And so we aren't going to say, you are the one that gets to do everything. And so this is a way to allow sort of, I think, participation.
It is in complete analogy to you not wanting to focus on one single pipeline to do VDJ calling.
Yeah, no, and I certainly absolutely see the benefit of having multiple different repositories and multiple different interfaces to the same to the same underlying data. I mean, certainly in the microarray world and a lot of other worlds that happens now. I guess I'm not so much concerned with there being multiple repositories of the processed data that give different views and different access policies and different features and stuff. I guess I'm not sure why they necessarily have to.
I mean, again, that's why, well, sorry, Felix.
Maybe it's a technical.
He's rolling his eyes, so we better stop. Yeah, I think that's the reason for having the registry, or, I mean, and we need to decide on what that is, but there may be some central access point where, I mean, I think it's different than having 5 places where an individual can go to each one individually versus saying, these are the 5 that are in operation, we want to put an umbrella over them, or, it may be that one repository can touch data from other repositories. I mean, again, we have to decide on what this is we're talking about, but that's where we're coming from.
This adds a layer of machine interoperability on top of the status quo in a certain sense. And then hopefully, you can imagine that over time, one of them will win and become the central one. It'll be mine.
We gotta give tools and resources a crack at it. Oh, you want to? Well, we could take you want a 10-minute break? Yeah, sure. 10-minute break at 4 o'clock. Tools and resources. Okay. Oh, there we are. Okay, great. Well, if we all get seated, we can let tools and resources piss everybody off and give them their chance. Eric.
Hello, everyone. So last session, hope you are re-energized. So Chaim Shrem and I have been leading the Tools and Resources Working Group. Chaim couldn't make it today, and so I was very lucky to recruit Christian up here with me on the podium. So we had a nice meeting this morning and have been chatting via video conference and working on a shared document, which the link is here. The other thing that has been going on is there's been a whole lot of discussion on this website, BTCR, that is a discussion site for repertoire analysis. And so although this was really heavily used for our working group, this is something that, of course, any discussion can happen on. So for instance, we've been talking about VDJ annotation formats and a lot of the issues that we're going to be talking about here. So our working document, oh dear, references this quite a lot. Well, okay, well, one moment. Okay, great. So I think the format that we'd like to try out is having 2 questions per slide. So any questions? No, just kidding. Okay, so this first slide should probably be fairly uncontroversial. We're hoping to promote transparent sharing of tools, methods, and information to be able to review, contribute, and continue development. We'd like to provide a platform for sharing current experiences with methods, their limitations, and emerging technical issues. We'd like to be able to discuss and communicate current challenges to ensure that tool and resource developers are focusing on the needs of users. So maybe I'll just skip over unless somebody has a burning question about these sort of guiding principles. Okay, so now, here we go. So we believe that software tools should use open source licenses. So in particular, we like the licenses that are approved by the Open Source Initiative, be hosted on publicly available repositories with versioning, use community-curated standard file formats, strive for modularity and interoperability with other tools, include example data and checks for expected output, provide information about run parameters as part of output, and clearly list hardware, operating system, and software dependencies, and or provide a script to build a virtual machine. So I would say that the second is definitely more best practices than the first. So in particular, Docker is a tool that is commonly used, and they have Docker files for describing how you can build a virtual machine. Another final point is it would be nice to clarify whether the software is still under active development, what level of support users can expect, and how to obtain support. Really, somebody disagrees with something on this slide. Danny. Oh. Okay. Well, I mean, there's definitely things that people are going to disagree with in later slides, so maybe we'll just skip ahead. Okay. So, there's lots of software tools out there, maybe too many, and we need some means of benchmarking them. So the 2 directions in which this can go is one is simulated data, and of course many people when they write a software tool also write a simulator. This is good. However, those simulations might be especially suited for the assumptions of that particular software tool. So we need to have some means of sharing simulated data and establishing certain sets of simulated data as being standards with which people can use to compare. Also, it would be great to decide on a collection of true data sets that we can use for cross-comparison. And we had a discussion about how there's lots of different dimensions that exist in real data. So for instance, different sequencing platforms, error rates, how many sequences there are, whether there's from diseased individuals or healthy, what species, and various cell types. Yes, and regarding simulated data, we also need summary statistics that can be used to benchmark the simulators. So I'll leave it to an exercise to decide whether we should have a benchmark simulator, benchmarker tool as well. Amazing. So the question was whether we should focus exclusively on human repertoires or on, do you mean species that use gene conversion as a special? Yes. Well, I would say everything. So it would certainly fall in the purview. Okay. So now on to slightly more the biological side of things, though of course computational protocols are very important. We believe that protocols should be made publicly available in full detail, and the criterion for that is that there is sufficient detail to be able to reproduce the entire experiment. So this of course goes substantially beyond what's usually described in the method section of the manuscript. Especially important is being able to see change history. So if you're updating protocol, it's important to know what parts changed. So there are specific mechanisms for doing that with version control, but it's just as effective to, when a protocol is changed, to add a line at the top that says this is what changed, when it changed, and how it changed. We believe that protocols should be kept in a versioned DOI granting repository and updated as necessary. And although we don't think that this hosting is something that we should get in the business of, we do think that we should pull together a central searchable location to list the available protocols.
Would you consider using something that's already out there, like protocols.io, for example?
Yes, we did have a vibrant discussion about protocols.io. So it's a beautiful piece of web software that allows you to upload protocols and version them and share them and have other people comment on them. We thought that it would be difficult to ask people to input their very complex 30-page protocols into protocols.io. Although there are ways of importing protocols, you can't just sort of plop your PDF or Word document in protocols.io and have it parse it. But it would be wonderful if we could go in that direction, for sure. So biologicals. Biological material should be available to interested researchers, and we recommend the use of commercial repositories for redistribution. And again, we don't want to get in the business of doing this distribution, but we believe that having a central list with the available materials should be maintained online by the community. I really think that people are going to have a comment about this. But so there's lots of different library preparation methods, and it's very difficult to benchmark them. So if it was possible to develop biological resources that would allow us to benchmark these library preparation and sequencing methods, that would be wonderful. So one sort of first-tier suggestion was, say we take just 10 stably modified non-lymphoid cell lines expressing either immunoglobulin or TCR sequence. So with the ideal that maybe in 5 years we'll be able to scale this up to the size of something that's more like a real repertoire.
I like the idea. Stable is an issue, I think, sometimes with the expressing cell lines. So someone's going to need to do a lot of work to try and make sure that they are stably expressing, because it doesn't always work that way. Sometimes they just go off after a few months.
Okay, I think this will always be an issue. The reason why we said that we would like to have a stably modified non-lymphoid is that probably the situation will get even worse if we're trying to resort back to hybridomas, and because then people might go back to the hybridomas that they had in liquid nitrogen for the last 15 years with unclear fusion partners and so on and so forth. So therefore, creating these things de novo should in the end be a workload that could be handled. But of course you're right. So I mean on the individual line has to be standardized before such a preparation would be made. In the end we're facing the problem that it's kind of a service to the community to prepare these resources. And right now we don't have any model how the lab that would be doing this could be compensated. So right now the idea would be to say, okay, you can order these cell lines, or these cell lines are deposited, for example, at ATCC. You can order them, you grow them up, you standardize their expression by qPCR, and then you can just mix them in a 1-to-1-to-1 and run them in your experiment.
Why not have a synthetic repertoire of just sequences that you publish, and then the lab can purchase, mix, and do it on their own?
That's a good point. So of course having free DNA would be much cheaper in terms of production, and it would be easier to generate higher diversity for this one. The discussion that we had this discussion and said that there is a clear advantage to have cells to start with, because this covers larger parts of the protocol that still might be an issue. If you think about template switching, when you're preparing your RNAs and so on and so forth, this all would not be covered if you're using double-stranded DNA. However, it would be up for discussion to say, okay, well, there are different libraries, so basically you can use a higher or a double-strand DNA library to start with that has a higher diversity. On the other hand, you have a rather oligoclonal cell-based library to cover other parts of the protocol. So this is not excluded. But if we think about, well, the first small steps to take, then this would probably be the first approach on standardization or validation.
So this one I'm sure is going to generate some discussion. So we all know and love the IMDT database. However, there are some things with which we are not completely happy. One is a licensing issue. So we believe that germline sequences should be made available with a license that enables further sharing and extension. And there should be a clear and objective system, including explicit criteria by which new germline genes are added to the database. And we believe that this should include a means of submitting germline genes inferred from high-throughput sequencing data. And of course, there needs to be some metadata about these various sequences, about how they were inferred and the level of evidence that we have for having these germline sequences. It is a bit of an issue, like, okay, even if we believe this, these additional things that we want, is it up to us to go about making a new database? Is this something that we believe that would be fundable through existing mechanisms? And how could we make this happen? Because, I mean, in human, perhaps the need is not so acute, but we have a lot of people working in other organisms, macaque, certain types of mice, for which the germline databases are really clearly insufficient. So something needs to happen. So if you look in the fine print of the IMGT database, it describes that there's restrictions on licensing. So, you know, the only examples that I know, for instance, is I believe it was IG Blast originally was distributing a version of the IMGT database along with their software tool, and they were asked to stop doing that. So that clearly, shows that there's some limitations on what's available. And I guess IMGT does get money for licensing this to commercial entities. So that's clear that this is not just sort of a passing fancy that they decided to put a specific license on the database, that it's something that is important for them.
Not that there's not enough work to do already, but I imagine that this would also require us to develop another set of schemas for representing a germline database.
Absolutely.
Yeah, look,
and now for someone who has actually developed a germline database.
Well, I'd like to return to the issue of licensing, and I think somehow we've got to resolve this. I can't believe that IMGT has a hold on a repertoire like this, and perhaps related to that point is I think it's important to have a fourth dot point highlighting the fact that I believe, as I'll discuss tomorrow, that so many of the sequences in the IMGT human repertoire are rubbish. And we can't only focus on putting new sequences in. We have to come up with a clean repertoire. Now, I mean, the sequences that I think are rubbish may have a continuing presence in our world, because I could be wrong. But at the very least, these sequences are dubious, and a very clear question mark needs to hang over them.
Yeah, in the Google Doc, we describe how we need, clear tiers of evidence for various germline genes, and if it was possible to, deprecate or sort of maybe not remove, but definitely, like you say, put a big question mark on certain genes, that would be helpful.
So in the spirit of proposals, one of the things we talked about under licensing was that quite a few of the tools that are commonly used today are available free for academic use, but they're not necessarily free for commercial use. So the Yale tools, for example, are licensed that way, and I think the Michael Shoegaze tools are as well, but I'm okay. Now, that might also be a solution to the IMGT issue. If IMGT is saying that they want to be able to sell their germline license to commercial companies, I think what we as a community might be saying is we did all the work to provide that germline library, and we want to be able to take it back and modify it and maintain it in different ways. But perhaps we don't object to, people exploiting it for commercial purposes. I don't know. Maybe we could, maybe we could resolve things with IMGT that way. We can get free use of it academically.
But I think so. The question is not only about free use for myself, but about whether I'm allowed to change it and to redistribute it. So these are different parts in the license, and right now redistribution is not covered. So if I package an IMGT library along in a virtual machine, I'm actually breaking the license.
In which case, we might be back to saying, that in some way we have to do a clean room re-implementation of the human germline library to get it away from IMGT. And in that case, I presume what we would want to do is hold on to the commercial rights of that, for us, and hope to capture their market.
Okay, but then okay, this is something one could discuss. On the other hand, I mean, from my personal perspective, it runs against the spirit. I mean, if we're claiming, okay, we want a free and open database that can be amended by everyone, the question is, it's hard for me or very opaque to me on how we can, whether we could get a lot of money from pharmaceutical companies to basically finance this one. There were people in the working group who said, well, that their employer is actually paying IMGT, but of course the amounts are unknown. If this is a huge amount of money that could finance that structure, then we could think about it. Otherwise, I think I'm a little bit against selling these things off, because then usually we would give preference to larger companies that simply have the cash and put a more heavy strain on startups. So I think just to make that thing, even if it's a free database for all, where's the issue?
It's a good point. But I do think there's a problem for us with tools in that, you know, a number of people are working with license arrangements like that right now. And, you know,
no, but it's not a problem. I mean, if the database has a higher level of availability, okay, of openness than the tools that other people have, then there is no compatibility.
So it's more of an issue with the database than it is with the tools. I can understand that. Yeah. But we may, in that case, somehow have to find the way of doing an
yes, of course. I mean, there, if we should decide that we have to re-implement this, then of course this has to be financed, and the question is either the taxpayer pays for it, and I think at the moment that this is a substantial amount of money that goes into it, publicly funded, it should be open, because otherwise it's kind of a just financing industry. On the other hand, if the money that one could get from commercial entities is good enough, okay, then one could think about such an agreement.
Perhaps I should make the comment that I made in the discussion a week or so ago. I don't quite know what IMGT has power to license. I mean, the sequences are there for everyone to use. So if it's the names, is it the names? And the names, I mean, they were granted the authority to issue names by the WHO, and I can't believe that the WHO would accept a system where that authority gives them the right to license material.
Okay, so I mean, I talked this through with the lawyers of our institute, so at least this is EU legislation, which is also what IMGT falls under as a French entity. The names are not protected because you cannot put you can only put a trademark on a name, which must be non-existent, which has to be linked to a product. Okay, so which doesn't exist in this case. So that is off. As we already discussed before, in the EU there is a special database protection clause which generates sui generis right, which is, so it's not directly a copyright, but it behaves like copyright. So the moment you're putting together a database that is a non-trivial collection of data, even if that is free, basically you're granted some kind of IP protection, and this is what IMGT could always rely on.
But, well, so what about V2? I mean, do they equally carry?
Theoretically, yes. But of course, that's
two licenses over the same repertoires.
No, let's put it this way: the one thing is basically the right that you are granted as the copyright holder or the holder of the protection of that intellectual property protection. You're still free to make the decision. Okay, I'm distributing this under an open database license, and it's free for everyone to use. Okay, IMGT made the decision to put on their license, which says, okay, free basically for, you can use it as an academic researcher, otherwise you're not allowed, or there is no general permission to redistribute or to use it as a commercial entity.
Can I just clarify that for just you're almost bang on. There's no protection in the underlying data that's in the database. It's the form and the structure of the database that's protected under European. So it doesn't protect against independent generation or creation of the data. So again, there's no formal IP in the underlying data that's in the database. I was just going to say the other, I understand it's a little bit different because here you're talking about data and the others you're talking about vectors and actual substantive issues with the ACTT and so the American Tissue Type and Culture, sorry, I forget the acronym, and AdGene. It is quite common practice in licensing to prohibit third-party distribution. And so both AdGene actually uses the Universal Biological Materials Transfer Agreement, so the NIH UBMTA, and it also prohibits third-party or onwards distribution. So that is quite common, but I understand where your issues arise if you're talking about software tools, because that's sort of contrary to a lot of practices in the open software movement. And probably also here where you're kind of trying to add value to data, but when you're talking about the materials, it's quite common to have restrictions on third-party distribution as well. And then the other issue is around it's also very common, including in your UBMTA, which underpins at least some of these issues, is to have that delineation between for commercial and for non-commercial use. The problem is that those boundaries between those two activities, as was pointed out before, are extremely blurry. So it's very, very, very difficult to have a clear delineation between what comprises commercial and non-commercial. And again, just adds another layer of complexity to any legal agreement. So that's why even the NIH steps back and says, we don't even want you using these formal, big, long contractual licensing agreements. We want simple conditions of use. So you want to think backwards to think, how can we simplify these licensing terms to achieve our purpose without making them any more onerous than necessary.
Okay, so but this is actually the reason. So for everything that is really biological material, so non-digital stuff, we're not going to have right now an own repository or anything or new licensing agreements. I think because there it is pretty clear that for the distributing lab, there is a cost for production and for distribution, and therefore it's actually the best thing, plus handling the MTAs if necessary, so therefore putting that one at a commercial distributor to take care of
They're also non-commercial distributors, right? They're not-for-profit organizations, the ones that you had listed up there. But that makes complete sense. Like it makes entire sense to give materials to a repository because of all of the costs associated. It's much more difficult to distribute than data. But I'm just saying that in general licensing terms in research, it is quite common, at least from the material side, to have non-third-party distribution. But I agree that you really want to think about whether that's necessary for data because it's so much easier to manipulate and play with.
Jason's been waiting patiently.
I'm a patient man. So just one thing to build on what Andrew said is that in addition to the namings, IMGT, like the value that they add, is also, is it short? There we go. That would be rude if I make it taller. Okay. So the value that IMGT adds is not just the names, but it's also the clustering into families and the numbering scheme. And I think if you need to escape their IP, then you're going to need to address that as well. I mean, I know that a handful of the University of New South Wales germline sequences actually don't fit into the IMGT numbering scheme. So, if you are thinking about, we need to re-implement what they've done, go back to the source material and rebuild the database with annotations and everything, you also have to address the framework, the CDR annotations, whether that's extending the IMGT numbering scheme or just redoing it or what. But it's another potential issue as you discovered in germline.
So maybe you can actually work together also with industry there. The companies that dehumanization of other species. And nowadays, when you want to assign an INN number, in 2014 the regulations have changed, and actually they address domain gap align IMGT tool to assess the humanness of these antibodies. Well, the regulation is still not very clear, at least to me, but these humanization companies also have to deal with it. So they have to go to IMGT to use the tool in order to assess whether the humanization grade that they deliver is sufficient to call the sequence humanized. Officially, they can't do that. They can't use IMGT. They don't get a license to do that kind of stuff and do the analysis and bring that to the client. So I can imagine that these companies are also interested into setting up an open database to do this kind of stuff.
Is this issue that you're just addressing the one that also was sent around by the Antibody Society a couple of months ago? Okay, so because there was a complaint of the Antibody Society, I think, end of last year that they were not happy with this scheme. Okay. Okay, I mean, I think for the human stuff we can think about this. Otherwise, I think it's important to see that that IMGT has been authorized by WHO to do this. However, this of course only extends to human genes. For the other ones, I'm not sure how many mouse geneticists are here, but parts of the naming scheme of IMGT are in gross violation of pretty much everything that mouse nomenclature should be, including allele naming. So I think this is also something that one could work on. So in the end, there are, besides the licensing issue and what the things that we just discussed, to have a clear and objective system, basically how we identify germline genes in the genome, how we assess their functionality, how we call the different regions, and that this, of course, has probably been painstaking work done by IMGT over the years manually, but by now, with a proliferating number of species that we are sequencing, these are things that first have to be operationalized so that they can run in code, and then for the last 2% that the code cannot handle, we probably still would need human curation. But by doing it this way, probably one could also circumvent the problem in being accused by IMGT that we simply copied their database, because then it's pretty clear, due to the presence of the code, that everything was recreated from free resources, free genome sequences, using the protocols as implemented in code. Okay, and then naming schemes, we've also discussed this briefly. This is of course something that is a big issue, especially outside of the community, because a lot of people have fallen in love with their VH169 or whatever segment, and it will be pretty difficult once the names change. Would change here. So this is also something that we would have to discuss, whether one would maintain kind of a legacy naming and then simply add new names, or whether one would go for a complete break and a renaming. Probably the one situation, or the one thing that I think would argue against maintaining the old names is if you think about the bad situation about mouse TCR gamma locus, which only contains 7 V genes and has 2 nomenclatures. Some of these, or the naming cannot be distinguished from each other without knowing which nomenclature you refer to, and you're still addressing wrong segments for them. So thereby having a naming scheme which is also visibly completely incompatible with the current naming scheme could be an advantage there. But of course, there is a substantial risk that we lose people outside of the community at that point.
Yeah, I'd just add to that I think that some of the strength of the IMGT nomenclature, in a sense, comes from, well, you know, as you say at the top of this slide, IMGT has delivered so much to so many people. It's provided very easy tools to use, and particularly people outside this room, if they don't have something easy to turn to, if they turn to IMGT, then the IMGT nomenclature and all that goes along with it will continue to almost control the field, despite the fact that we here might think that we know more about it. If you've got hundreds of users continuing to use IMGT utilities because they're easy, we won't be in the game.
Yeah, sure. The problem, I think, with the general nomenclature, I think this is what most people by themselves, or the conclusion that most people by themselves have come to, is that having a numbering scheme that tries to capture the structure of the locus is fundamentally flawed. Okay, this works very well if you have one defined mouse strain like B6. Okay, then the numbers will always stay the same. Once you're running across different genotypes and across different alleles, then of course you will see duplications, you will see deletions, and especially the duplications will be pretty hard to annotate, because then your system breaks up since your numbering doesn't fit anymore. So therefore, I mean, other databases like VBase, for example, simply come up with a serial numbering, which then, okay, you have to find ways to avoid duplication. But in the end, I mean, this is a general issue that will have to be resolved independently of all the other things that we are discussing right now.
But just to stress the point that you made earlier, speaking as someone old enough to know the nomenclature and have worked with it pre-IMGT, that there's a lot of people out there that when they're doing their PubMed searches, they're looking for similar genes, and it's very, very difficult to trace back through histories of literature what you're actually looking at when the name of the one particular gene you're looking at has changed 2 or 3 times. So if some kind of compromise could be made so you can still retain the essential bit that you would use in a search engine and then put whatever else you want on after it, that might be acceptable to everybody outside who's used to their, as you say, pet genes like 169.
Yeah. Well, I think there are, I mean, if one would construct a de novo nomenclature, then probably one clearly would start with having locus and segment nomenclature in there, like it is now for the human genes. We would not go down that insanity like it is for mouse V genes for heavy, okay, where you're not even recognizing whether it's V or J coming from the name. So I think that's pretty clear. The big question is really how you do numbering and whether you would like to encode family membership, and as I clearly said, I think trying to encode position in the locus is probably always a bad idea, but as it might change. Same thing goes for trying to encode functionality, because the functionality of that allele that you're assessing in one strain or one haplotype might be different than the other one. So therefore, no pseudogene or whatever is in there.
So just to that point, it's you know, PubMed now has a capability to do synonyms behind the scenes. I know that, like everybody else, they're kind of overworked and have too much to do, but I think it's not beyond the realm of possibility for a group such as this to make a request if and when we come up with a different terminology to say, look, behind the scenes, can you make one of these things look for another? It's something to consider.
So that basically,
And they're nice. I mean, I've met those people.
which would basically mean that one would try to assemble a complete lookup table of all nomenclature schemes and then simply submit it and ask them to implement it in their search.
Yes.
Just as a final note, we have a lot of sentences that say something like a central resource with blank should be maintained online by the community. This is not a trivial task. I mean, even if it's just a collection of links or a table or something like that. So now we have a discussion forum, BTCR, but should we also try to centralize around some sort of wiki or something that we can all use for getting together and organizing things, and how can we incentivize people to contribute? So Jessica described how in the Rosetta community they give a cash prize every year for the person who's done the most to contribute to public resources, or answer the most questions or something like that. Answered the most questions. That sounds lovely. But you know, dollars or euros aren't necessarily the coin of the realm when it comes to academics anyway.
I would just move for GitHub as a common platform, especially if a lot of some of the things that we're maintaining are machine readable.
The only thing that because we did talk about this, and the only thing that we thought something like Wikibooks might give us is that discussion page that goes behind the wiki page itself. I'm not sure that's in GitHub.
So GitHub supports wikis in general, but also I guess it really depends on the particular type of tool, but for example, pull requests and issues are places that support lots of discussion. And for example, anytime there is some kind of diff that is merged in, the associated pull request should have all of the discussion associated with that particular issue.
And let's talk about it offline.
And we do already have a discussion forum.
Yeah, and then Discourse is also great, by the way. There are some plugins that do lots of statistics, and if you want to incentivize all kinds of weird ways that people contribute, it would be very easy to implement there. And it's pretty.
Okay, great. Well, I'm sure we'll discuss these things more. Thank you.
You see what I mean by the three groups having done a lot of work. It's been pretty amazing for the last year. And we've mentioned a couple resources and links. IRMAX, the group that's helped keep these meetings together, I sent out yesterday, it's AIRR 2016 to IRMAX website, and all the links. We'll make sure all the links are up there, and they were in the email I sent out yesterday with the wrong address. But that IRMAX site, just for now, so that there's a place you can go and find everything, that all these links, like the GitHub link, and then there's BTCR is pretty easy to find. And if anybody can't find these things, just email me and I'll make sure you get them. And so then I don't think I mentioned, so the demos start at 6. Well, the reception starts at 6, and there are appies, and there's going to be a lot of appies, so that's good. The demos last year at the 2015 meetings, they were like most fun, and kind of the time we got to most talk science, really, was that people got to show their science, that they're... Now I know, and cash bar, bring your cash. I only get like 10% on each drink. So. So that starts at 6. The demos start at 6:30, And then tomorrow morning we start out at 8:30 here. Well, 8 o'clock we'll walk a group over, but I think you guys know you can make it there. 8:30 we start with the intro to the day, and then at 8:45 we have our keynote. So we've got a big day, and then I've emailed people that have posters tomorrow. There's a poster session tomorrow and a poster session on Wednesday, and try to get your posters up. They're downstairs. The boards are there, and then at lunchtime you can look at the posters, and there'll be more time for discussion. But I just wanted to remind people that we're in poster session 1. That's tomorrow. Bring your posters at the beginning of the day, and you can put up the posters. So give ourselves a great hand for a good day and a lot of work. And see you at the demos.
Yeah.